नमस्ते। ओम सर्वे भवन्तु सुखिनः सर्वे सन्तु निरामयाः सर्वे भद्राणि पश्यन्तु मा कश्चित् ओम शांति शांति शांति।
Thank you, sir, for all of you. An institution is identified by two things. One is the success of its students and by the inspiration of its educators. An institution is set by its educator not only by impacting knowledge and mentorship. Our speaker is one such person who is such an academic leader who has dedicated his career to advancing the pharmaceutical education and research. It's an honor to have Professor Dr. C.H. Niranjan Patra sir as our resource person, a man of very few words but a man of impact and ever-ending charm. Dr. Niranjan Patra sir is currently serving as the Vice Principal Professor and Head of Department of Pharmaceutics at Roland Institute of Pharmaceutical Sciences, Berhampur, and alumnus of the College of Pharmaceutical Sciences, Mohuda, and Roland. He earned his PhD. Over the years, Dr. Patra has established himself as a prolific researcher and mentor. He has had 10 PhD scholars, authored 1 Indian patent, 4 books, 2 editorials, more than 70 research papers in different national and international journals, and 10 review articles, with an impressive X index of 25, reflecting the quality and impact of his research from UGC, AICTE, BBUD, and his election as a fellow of Institution of Chemists. He is also a life member of IPGA, FDP, IPGA, IPGA, and IPGA, demonstrating his active engagement with the professional pharmacy community. Today, we are privileged to learn from his rich academic experience and research expertise. Dear faculty members and delegates, let's extend a warm welcome to our research Dr. Niranjan Patra sir with a big round of applause.
A very nice and short introduction about our research person. And I would like to announce Dr. Prasad Kumar Chaudhary sir is the chairperson for this session, and now will start this session. Now it will go back to our research person, Dr. C.H. Niranjan. Thank you. Have a nice day. We started it very happily. Thank you. Jai Jagannath once again.
Good morning, dear friends. At the outset, I would like to thank the principal of this institute, Dr. N. Usharani Madam, Vice Principal, Dr. Uma Vishnu Sir, the organizing secretary, Dr. Ranjan Mahapatra, all other faculty members of this institute, and all the participating faculty members. Thank you very much for giving me this opportunity. I hope I will meet your expectations. I would also like to thank the chairman of the session, Dr. Prasad. My topic of presentation is lipid-based nano formulation, scientific insights, and some case studies. So, in my presentation, I will be focusing on the criteria for selection of drug to formulate a lipid-based nano formulation, the scientific insights regarding the selection of oils, selection of solid lipids, selection of liquid lipids, what should be the ratio, all those intricacies will be discussed, and at the end or in between, I will be showing some of the case studies from the research work that we have done. Oh, negative transfer. Negative transfer देवे. So let me continue the presentation. So according to a research publication in the year 2025, around 60 to 70% of API belong to the BCS class 2. That is, they are poorly soluble and highly permeable drugs. So there is a need to develop a drug delivery system which can overcome the challenges that are generally associated with BCS class 2 drugs. Overall, there are 4 different classes of drugs according to BCS: class 1, class 2, class 3, and class 4. So the class 1 is, as you all are faculty members, you must be knowing, class 1 is highly soluble, highly permeable. Class 2 is poorly soluble, highly permeable. Class 3 is highly soluble, poorly permeable, and class 4 is poorly soluble and poorly soluble. Just I would like, as Anjan sir has told, make it interactive. Can just someone tell that what is the meaning of highly soluble and highly permeable? Any of the participating faculty members? Anyone would like to share? When we say that highly soluble, what is that? Yes, please. What is soluble? So that we can say. So there is a official criteria to say whether a particular drug is highly soluble or highly permeable. The highest dose of the drug should be soluble in 250 ml of water in the entire pH range of 1 to 7.4. That is in the entire gastrointestinal pH range. So if a particular drug is available in 2 or 3 different doses, the highest dose must be soluble in 250 ml of water, that too in the entire pH range. Suppose a particular drug is soluble in the stomach fluid but not soluble in the intestinal fluid, it will not come under the category of highly soluble. It has to be soluble in the entire pH range. Similarly, to say a particular drug is highly permeable, 85% of the administered drug should reach the systemic circulation. Under that condition, we say that a particular drug is highly soluble. So this is my topic of presentation. As I have just told you that 60 to 70% of drugs that are in the pipeline to come to the market belong to the BCS class 2 drugs. And this is the definition, that is highly permeable means when more than 85% of the administered dose is absorbed or reaching the systemic circulation, whereas highly soluble means maximum dose should be soluble in 250 ml of aqueous medium. Okay, so this is the definition. So in my presentation, I am focusing mainly on PCS class 2 drugs. So there are various approaches by which we can improve solubility of a drug. Like we can go for a prodrug approach or particle size reduction like micronization or nanonization, converting a drug into salt. Can use co-solvents, can go for complexation with beta cyclodextrin, HB beta cyclodextrin, solid dispersions, liquid solids. But mostly in all these cases, they improve the solubility of the drug, but they do not increase the permeability to a significant extent. That's why I have focused on the lipid-based formulations, which have the unique ability of enhancing the solubility, dissolution, absorption, and bioavailability. So now the question comes, how this idea came that a lipid-based drug delivery system can be developed. So according to a study, halophantine, when it was taken in empty stomach, its Tmax was around 184 and AUC was 3.9. But when it was taken along with fatty food, it has shown a improvement around 8 times in case of Cmax and around 4 times improvement in AUC. So in presence of fatty food, some drugs, those are generally hydrophobic drugs, they show a significant increase in the bioavailability. So the idea came, why not to use fatty substances or lipids in our drug delivery system, so that even if the patient has taken fatty meal or not, the drug will exhibit a good bioavailability. So based on that, the idea came, and the reasons for improvement in bioavailability are when we take a drug along with fatty food. These are the different reasons. There will be an increase in the bile flow. There will be increase in pancreatic secretions, which helps in micellar solubilization. It increases it enhances the lymphatic transport. It increases intestinal wall permeability and reduces P-glycoprotein efflux activity. So details I will be discussing in my coming slides. So these are the different reasons why a fat-rich food increases the bioavailability of drug. And because of these findings, FDA has made an observation which led to the development of guidelines established by US FDA. That is, one study is mandatory. If it's a new chemical entity, they must perform impact of food in the enhancement of bioavailability. And if it's a generic drug for ANDA approval, they must study the fed bioequivalence study. So the effect of food in presence, bioavailability of drug in presence of food. So now coming to my topic, that is lipid-based drug delivery system. So in the case of lipid-based drug delivery systems, the drug is either dissolved or dispersed in a lipid excipient. Mostly these are the components of a lipid-based drug delivery system. One is lipid, second one is surfactant, and third one is co-surfactant. So there are several forms of lipid-based drug delivery system. Like it can emulsion, which utilizes oils. It is also coming under lipid-based drug delivery system, like microemulsion, nanoemulsion, lipid particulate systems like lipospheres, SLN, solid lipid nanoparticles, nanostructured lipid carriers, and the vesicular systems like liposomes, neosomes, kethosomes. In my presentation, I am focusing on self-emulsifying drug delivery system, solid lipid nanoparticle, and nanostructured lipid carriers. So when we use lipids or oil in our formulation, it is showing two major effects. One is enhancement in drug solubility, and second one is favorable effect on lymphatic transport. So when suppose we make a formulation which contains drug with some oils or lipids, when it is taken orally, the fat goes to the intestine and it sends a signal to the gallbladder, which makes the secretion of a digestive juice, which is called as the bile. So bile salts, and simultaneously there is release of lipase from the pancreas, and this pancreas will convert the fats into fatty acids and monoglycerides, and these monoglycerides, fatty acids, bile salts, they form mixed micelles, which is helping in solubilization of the drug. So that is one of the major mechanisms of enhancement in solubility. Second important one is favorable effect on lymphatic transport. So the triglycerides that are present in our formulation, they will be forming a larger moiety which is called as chylomicron. So the chylomicrons are larger moieties which contains the drug. Because of their larger size, they are unable to penetrate into the blood capillary. But they can go through another route that is called as lymphatic capillary, which is lacteal. There, the pores are of larger size, and the chylomicron is able to pass through the lacteal and then it goes to the systemic circulation. So it has two advantages. One is through the lacteal, some amount of drug reaches the systemic circulation. Second one is whatever drugs that are entrapped into the chylomicrons, they cannot enter into the blood capillary. If the drug goes via the blood capillary, then it also has to expose to the portal capillary. So there will be loss of drug because of the fast metabolism. So these things make these lipid-based formulations unique. They enhance the solubility. They also enhance the bioavailability to lymphatic absorption. So this is was the introduction part. Now, as I told you, I will be focusing on self-emulsifying solid lipid nanoparticle and nanostructured lipid carrier. So what is a self-emulsifying drug delivery system? It's an isotropic mixture. It's a single phase mixture of oil or mixture of oils, surfactant, and co-surfactant. And when it comes in contact with the gastric medium, it forms a emulsion which is called as oil water emulsion. And depending on the globule size, we say it as a self microemulsifying system or self nanoemulsifying system. So self-emulsifying drug delivery system has also been commercially successful. One of the drugs that is called as cyclosporine A is launched by the German-based pharmaceutical company Sandoz, and some other brands are also available which are formulated as self-emulsifying drug delivery system. So this drug delivery system is not only limited to academic research; it is also successful when it comes to commercialization. So when we are selecting oils, in the case of self-emulsifying drug delivery system, there are three important components: oil, surfactant, and co-surfactant. So if the type of oil is a long hydrocarbon chain or long chain triglyceride, it will have a very good ability to increase drug transport through the lymphatic system, but generally they form emulsions in the micron range. Whereas the case of medium chain oils or triglycerides, they form globules in the nano range, but lymphatic transport is relatively less. These are some of the examples of oils that are used in formulation of self-emulsifying drug delivery. So the second important component of this is surfactant. So how to select a surfactant? So the first important point is the surfactant showing maximum ability to solubilize the drug should be selected as the surfactant. Generally, surfactants having HLB greater than 12 are helpful in getting emulsions in the nano range. And the concentration of surfactant also plays an important role. It should not be beyond 60%. It has to be used within 60%. And these are some of the roles that are played by surfactant in the self-emulsifying drug delivery system. That is, they reduce the surface tension. It forms a monolayer between the oil phase and aqueous phase. It inhibits the P-glycoprotein efflux transport. One of the success in the case of Accredited. Accredited was formulated with polysorbate 80, and the polysorbate 80 inhibited the PGP efflux transport, and because of that, the bioavailability of Accredited was low. And surfactants increase the permeability by distributing the structural organization of the disturbing the structural organization of the membrane. And these are some of the surfactants that are used. You can see the lower part. All the surfactants having HLB greater than 12 are suitable for self microemulsifying or self nanoemulsifying drug delivery system. And third important component is the post-surfactant. It helps to increase the drug loading capacity. It reduces the local irritation, and sometimes instead of using a single surfactant, we can go for a combination to reduce the local irritation. And these are some of the examples. Okay, now I would like to present a case study published by one of my research scholars, Sri Kanucharan Panigrai, in Journal of Drug Delivery Science and Technology. We also have made some other publications in self-emulsifying and nano-emulsion. So in this particular case study, the first criteria is selection of drug. What should be the criteria for selection of drug? So the water solubility or the aqueous solubility of the drug is very low, 0.0094 milligram per ml. Its dose is moderate, like around 62.5. Its bioavailability is 50%. Lipophilicity, one of the important factors when you are selecting a drug for self-emulsifying drug delivery, it should be at least around 5. If the log P is around 5, it's a suitable drug to formulate a self-emulsifying drug delivery system. It's a BCS class 2 drug, and it belongs to the therapeutic category, that is in the case of pulmonary hypertension. So because of these features, this drug fits into the self-emulsifying drug delivery system. Suppose the dose would have been higher, like 400 or 500, then it is always difficult to formulate a self-emulsifying drug delivery system, because you have to encapsulate the entire dose with oil, surfactant and post-surfactant, so that total quantity will increase, and if you are further solidifying it, the dose will be, means the total weight of the tablet will be very high. So one of the limitations you can say for self-emulsifying drug delivery system is dose should be lower so that you can proceed with self-emulsifying drug delivery system. So the first study is screening of excipients. You have to screen oil, surfactant and post-surfactant, and after screening we found that these are the selected excipients. Kepmol MCM is selected as oil, Labrosol as surfactant, and PEG as post-surfactant, PEG 600. Then the next step is we have to proceed for construction of pseudo-ternary phase diagram to find out the optimum mixture of S-mix, oil and water. So S-mix means it's a mixture of surfactant and post-surfactant. We have prepared 3 S-mix, 1:1, 1:2 and 2:1. Then it was mixed with the oil in different ratios, and it was titrated with water, and we have determined the amount of water required to get to see the clarity, time for self-emulsification and dispersion. Then we got these ternary phase diagrams. So the diagram whichever is showing higher emulsion region is selected. So in our study we got good result with 1:1 ratio. Then we have started the formulation. So whenever we make a formulation, we need to have some targets in our mind. So these are our targets. We wanted that Q15 should be more than 90%. That means more than 90% of drugs should be dissolved within 15 minutes. The globule size of the nano emulsion that will be formed after the agitation should be less than 100 nanometer. PDI should be less than 0.3 so that it will have a uniform size distribution, and emulsification time should be less than 2 minutes. So this is our target, and by keeping this target in our mind, we have prepared the formulations in which two important factors are selected. Oil is one of the important factors, and second one is the S mix, mixture of surfactant and co-surfactant. And these are the concentrations. So here you can see that Kepmol, as the concentration of S mix increase, the drug release rate increased, and as the concentration of oil increased, drug release rate decreased. So surfactant or the S mix has a positive effect on the release of the drug. Similarly, globule size decreased with increase in the concentration of S mix, and as the concentration of S mix increased, the PDA decreased. So these are some of the evaluations. Like we also perform zeta potential. Usually, if the zeta potential value is plus 30 millivolt or minus 30 millivolt, it is accepted as stable. But we could not get in that range. Our best for we selected run 4 as our best formulation, but it was around 26 minus 26.8. But it is theoretically it should be more than 30. And we, at the end, we performed a pharmacokinetic study which showed a 2.5-fold increase in the AUC of Ozempic. So this is the conclusion. So by formulating this, we got a 2.5 times improvement in the oral bioavailability of Ozempic. Then next comes to the solid lipid nanoparticles. So let us first discuss about theoretical aspects. So solid lipid nanoparticle consists of a solid lipid, means a lipid which is solid at room temperature. Usually the size range of SLN is in the range of 50 to 1000 nanometer. Apart from solid lipid, we also have to use surfactants and co-surfactant. As you can see here, the central core is the solid lipid, and if our drug is soluble in the lipid, we have to dissolve the drug in the liquid, and then we have to mix with surfactant and co-surfactant so that it will be forming a monolayer between the liquid phase and the aqueous phase. So in the previous case, in case of self-emulsifying drug delivery system, mostly it is suitable for achieving a quick release. But in this case, as we are using solid lipids, and that too, those all these solid lipids have a low HLV value, so they are more suitable for controlled release or sustained release. Okay, so these are some of the advantages and disadvantages of solid lipid nanoparticle. We can prepare solid lipid nanoparticle for different applications. We can use it for protection from light, protection from moisture. We can prepare it for improving the overall bioavailability of a drug. And one of the major advantage of this technique is it is scale up is possible. Even at the lab scale and also the industrial scale, it can be used. We can avoid use of organic solvents. That is another major advantage, and it is suitable for controlled or sustained release. And the disadvantages are the drug loading is less. So if the drug loading is less, it will not be suitable for high dose drugs. It may be a very good approach for low dose drugs because of the reasons like the lipids. If the lipids remain compact, there is also a chance that expulsion of loaded drug, even if it was incorporated into the lipid, after some time again it may come out of the system. And sometimes, as these lipids are used, storage may lead to polymorphic transition and particle use. Polymorphic transition from alpha form to beta form, so it becomes more stable, then it leads to expulsion of the medication. So, as the important components of solid lipid nanoparticle is lipid, surfactant, and co-surfactant. So, the lipid means here it's a solid lipid, whereas in the previous case it was a oil. So, first requirement is melting point should be above 40 degree. It should not be below 40 degree, because once it is administered in the body condition also, it should remain a solid. Second is, whatever solid lipid that we are using must have high affinity of drug for lipid phase than the aqueous phase to prevent partition. So the drug must have a high affinity for the lipid phase. Then only it will remain in the lipid phase. It will not partition out to the aqueous phase. And the hydrocarbon chain length, if it is of higher long, it's a long chain, it results in formation of particle size, higher particle size, and prolonged release. And this last point is very, very important. That is lipid polymorphism of crystalline. Suppose in our formulation we are using pure lipids, means it's either it's not a mixture, it's either a pure triglyceride. Then in that case, what happens? You can see the first part, the arrangement. It's very, very compact. So there is no space for entrapment of the drug. And even if some amount of drug is entrapped, there is a chance that it may expel. Whereas the second one is a mixture, complex lipid matrix, which is a mixture of mono, di, and triglycerides, like pompridol is one example. So here you can see different types of lipids are used, mono, di, and tri, and because of that, there are a lot of spaces available, and more drug can be entrapped into the lipids. So it is advisable to use a mixture of mono, di, and triglycerides rather than using pure lipids or one type of lipids. And these are some of the examples of solid lipids that are used in SLN. So you can see almost all are having a higher, means more than 40. In only one case is there 30 to 35. If you are going with SLN, it is going to show a failure. And the HLB value also, if you will see, all are at a lower level. Because of this, it's very suitable for sustained release. So this is my second case study. Lyophilized SLN of sinacalcet hydrochloride for in drug development and industrial pharmacy. So in this case, we have used another drug that is called as sinacalcet hydrochloride. So here the drug is poorly soluble. If it is less than 10 milligram per ml, we can say it's poorly soluble. Here it is 1.28 milligram. It is available in 3 different doses. Its bioavailability is low, 20 to 25%. Log P is perfectly alright for making SLN because it is more than 5. It's VCS class 2 drug, and it's used for hyperparathyroidism and parathyroid calcium. So because it fits to the requirement, we selected this drug. Then preparation of solid lipid nanoparticles. So there will be a liquid phase and there is one aqueous phase. So in the liquid phase, drug, solid lipid were dissolved, and simultaneously there is one aqueous phase in which we have dissolved the chloroxamine in water. Both are heated to 80 degrees, then they were mixed. The pre-emulsion is formed, then it was sonicated for further reduction in the globule size, then cooled to room temperature to get the SLN. So here also we have to set a target. So these targets we have set. Entrapment should be 70 to 100, entrapment efficiency. In case of solid lipids, it is always very difficult to get particle size or globule size less than 100. That's why we have set a higher range. In case of self-emulsifying, the target set by us was less than 100 nanometer, but here we set a target of 200 nanometer. I have written here 10 to 200, but it can be 1 to 200 also. But practically, it is very difficult to get that level. And T85% means time taken for 85% of drug release, and we have fixed a range that it should be in the range of 18 to 24. This is our target, and based on that, as per PBD design, we made a formulation, 17 formulations, and these are the results. And we found that run number 10 meets our target. Like you can see, the entrapment efficiency was in the range of 70 to 100, and then the particle size was less than 200 nanometer, and time for 85% 85% drug release is 20.2, which our target was it should be 18 to 24. So we selected this formulation. Then further we lyophilized it by using mannitol as a cryoprotectant, 10% weight by volume, and these are some of the evaluation tests. So that is a sustained release for 24 hours in case of SLN as well as in case of lyophilized SLN. And then this is another interesting finding, that is from the BSc study. The pure drug exhibited a sharp peak at 181 degrees centigrade, indicating it's a crystalline drug, whereas when it is formulated as a lyophilized SLN, that is F, it is not showing any peak. This may be due to molecular levels, dispersion of the drug in the molten liquid. We have also performed BSc, XRD. So in the XRD study, we got peaks at different 2 theta angles, but in the case of formulation, that is C, the number of peaks reduced drastically, indicating that there is partial homogenization of the drug in the lyophilized. Then we have gone for the bioavailability study, pharmacokinetic study by Latin square crossover design. So we have selected 6 animals, and it is divided into 3 different study periods. The 3 samples that were given, one is aqueous suspension of pure drug, one is SLN formulation, and third one is lyophilized SLN. And here you can see there is a significant increase. There is nearly 2 times increase in the AUC for both formulations were observed. So this is the conclusion, that 2 times improvement in bioavailability. Next is NLC. So this is NLC. So the difference, this is called as second generation of lipid nanoparticle. SLN is called as first generation. So here the only difference is we are incorporating one more third component, that is called as liquid lipid. Apart from solid lipid, we are also adding a liquid lipid, and surfactant will be there. So as you can see here, the solid lipids are the green rectangles, and there are some liquid lipids which helps in creating more space for accommodating the drug. So rationale for shifting from SLN to NLC. So here you can see the first part of the diagram. Here it is a compact structure, and because of that, drug entrapment is less in case of SLN. But in the case of NLC, due to the incorporation of 20 to 30% of liquid lipid into the solid lipid, it creates a lots of voids or spaces into which we can entrap the drug. You can see the second part of the diagram, which is the NLC. So the perfect crystals, what happens in the case of perfect crystals, they are highly ordered. So they are very closely packed, and that do not allow the entrapment of the drug. Then some other important criteria for selection of solid lipid, as in the previous case, here also melting point should be greater than 40, polymorphic behavior. So here also again you can see, if you are using different types of liquids, then what happens? It is leading to the formation of a lot of space will be created and you can accommodate the drug. Whereas if you use a single type of pure liquid, it is first one is the compact form. So we need to use, so by in the previous case also I told you that if you are using a pure lipid, space is less. But when you are using a combination of mono, di and triglyceride, more space is available for entrapment of drug. But when you add further one more liquid lipid, it still increases the entrapment efficiency or drug loading capacity. Similarly, another important parameter is as we are using lipids, lipids generally has a tendency to convert to the stable form, that is the beta form. Beta form is more compact, but what we want, it should remain in alpha form or beta dash form. So for that, what is important? We need to control the cooling process. We need to control the different parameters in such a way that there is no conversion to the beta form. It remains in the alpha or beta dash form, so that more space is there. That the upper part of the diagram, which is suitable, because and that is possible because of controlled cooling. And the second important component is liquid lipid. So liquid lipid must be soluble in the molten solid lipid. Otherwise, it will lead to phase separation. Drug must be highly soluble in the liquid lipid. And how much liquid lipid we can use when we are making a NLC. So the ratio is 70 to 30 is the maximum. Means 30% should be the maximum quantity of liquid lipid that should be used. Suppose you are going beyond that. If you are increasing 40% or 50%, there is a chance that it, instead of getting a nanostructured lipid carrier, you may get a nano emulsion. So when you are getting a nano emulsion, it is going to change entirely the pharmacokinetic parameters. NLC will be giving you a sustained or controlled release, whereas if you are increasing the liquid component, there is a chance that it may become a nano emulsion. So we have to take care, and these are some of the examples of liquid lipids. And this is just similar type. So it's a BBD enabled NLC of cinacalcid hydrochloride. We have also some other publications in cinacalcid hydrochloride. We have lyophilized that and studied its physics. So this is the same drug. So I just don't want to repeat this. It's the same drug. Earlier I have made SLN. Here it is NLC. When it was made into NLC, the advantage was, so with the same pattern we have gone, we have evaluated the parameters. Pharmacokinetic study showed nearly 4 times increase in the AUC. When we made SLN for the cinacalcid hydrochloride, we got 2 times increase in the AUC, whereas in this particular case, we got nearly 4 times improvement in NLC. Okay, so NLC formulations are superior compared to SLN, and we can get a better bioavailability. Finally, I would like to conclude by saying that these lipid-based drug delivery systems are easy to manufacture and scalable. They can be manufactured at larger scale, suitable for improving the solubility of hydrophobic drugs with the help of surfactant and co-surfactant. It improves the absorption by lymphatic transport. So a careful selection of oil, solid lipid, liquid lipid, surfactant, co-surfactant plays an important role in the successful formulation of lipid-based nano formulations. These are my references. I am thankful to all my research scholars, PhD and MPhil. It is their hard work. I am just presenting this. So it's
Good morning, my Dr. here. So it's actually really, it's my privilege and I am really happy to be a chairperson of the station of my Professor Sir, who happens to be one of my seniors and my teacher when I was a student at CPS Mohara. To start with this topic, very aptitude by Professor Sir, we we have FDP is designated for this basics and advancements in, of research. So particularly that theme has been covered by our research person. He started with very basic note about how this solvent enhancement becomes a very challenge for everyone, pharmaceutical persons, and how to develop the solubility. And these research explain an interest in development of nano-based lipid formulations, and it is a very demanding research area, particularly for those of us who are mentally the students or research work and research analysis. I hope this presentation, which was very insightful and comprehensive, might have given so much idea and scope for guiding upcoming researchers. Thank you, sir. On behalf of the organizing committee, royal family members, and the members of the Indian Pharmacy Graduate Association, I convey my heartfelt thanks to you for accepting our invitation and putting this insightful and comprehensive topic, lipid-based nanoformulations, emphasizing on the self-diversifying religious systems, and also stating with us your expertise research among all our members who have joined this activity. Thank you. Now, though it was very, you have observed the slides very simple and very schematically presented, but still, if there is any participant who has got any query, you may interact directly with our research person. Please.
Sir. So, regarding the translation from unqualified form, how often do we use ES versus XR for stable testing? Yes.
It's a very important question because one of the major problems associated with all such things is that lipids generally have a tendency to get converted into the beta form. So this problem is not observed at the time of manufacturing. It is generally observed during the storage stability study. So in such cases, we have to do the stability studies, and every 1 month interval, we have to check what is the in the DSC or XRD, and is there any change in the polymorphic behavior or not. It has to be checked. So it is generally performed during the stability studies. Another problem is that even if the drug is, because when we make it, we dissolve the drug in the liquid, so it is expected that it will remain in the NLC or SLN. But because of this conversion, it will be inside. But when it gets converted to the stable form, it gets squeezed or it becomes more compact, and that time there will be expulsion of the drug. So we may be thinking that we have a very good SLN or NLC with entrapment efficiency 70% or more than that, but with time there may be a decrease in the entrapment. Entrapped drug comes out of.
Any more questions?
Thank you, sir, for a nice presentation. I have a simple doubt. No doubt, lipid nanoparticles are the recent advancement in drug delivery, stability, portability, availability enhancement. I have a simple doubt that we are using lipids. Whether these lipids have any effect on lipid profile of the patient to the hyperlipidemia, or I don't have a specialization in pharmaceutics, but a question come to my mind. So since we are using lipids, whether it will affect the lipid profile of the patient?
This is really a very good question, and it is expected from a scholar like Dr. Sudhir only. So definitely, when we are using lipids, it may have effect, but the concentration or the quantity that we are using may not have any, because there are no such reports that if you are using SLN or NLC, it is going to increase the lipid profile or the cholesterol level or
So
those.
whether there are any studies regarding to this, or it might be open to study.
I think it is.
can be extended.
Actually, I am not very sure whether there are any reports related when you are using lipids in your formulation, what are its impact on the patient. There must be some studies, but right now I do not have that.
That is.
Important now. We extend it about half of the
So that we need to have a we need to use ProSim ternary phase means ProSim software. Here, what I will explain you simply. Yes. So yes, very good. It's important because that is the basis on which you will develop your formulation. So first component is S mix. We are using a mixture of surfactant and co-surfactant. So in our case, I am just giving you 1:1. Like suppose you are taking 5 ml of total surfactant. In that surfactant will be 2.5 ml and 2.5 ml of co-surfactant. That is first component. So that 5 ml is fixed. Next component is oil. What we will be doing is oil will be increasing the suppose 4.5 ml of S mix. We will mix it with 0.5 ml of oil. So total is 5 ml. Similarly, next one is 4 ml of S mix, 1 ml of oil. 3 ml of S mix, which is a mixture 1:1, and 2 ml of oil. So that mixture will be prepared like 2 ml of S mix and 3 ml of oil, 1 ml of S mix and 4 ml of oil. This mixture is ready. That we will take in a conical flask. Then we will, in a burette, we will take water, and we have to put it. We have to put drop-wise water. It's a titration. So the amount of water required to form a clear emulsion, we have to observe it visually. So the point at which it forms a clear emulsion, we'll note the amount of water consumed. Then now we'll get the percentage, like what is suppose 4.5 ml of S mix, 0.5 ml of oil, and amount of water consumed for getting a clear emulsion is 2 ml. So those three quantities we have to convert this into percentage. Then we have to put it in the ProSim ternary or ProSim software. Then when you put it, it will be displaying the emulsification regions. So more the emulsification zone or more the emulsification region, better is the composition. So we have done it with 1:1 of S mix. Similarly, one more with 1:2. Like if it is 4 ml of S mix, means one part is surfactant and 2 parts of co-surfactant. And again, we have to change the oil quantity. And we will do so whichever S mix is showing better emulsification zone, or higher zone, that should be selected as for further formulations. Okay. So that the software is free. Anybody can download and use. ProSim.
Thank you so much.
One is surfactant is one of the formulation. What should be the concentration of surfactant? Are we going to observe everything when the emulsion is going from the acidic environment of stomach to the
So the first point is surfactant. There is a limit. You cannot use beyond 60%. That's the limit. Or sometimes, if we are not achieving our objectives, we can go for co-surfactant is not must. We can sometimes we go for the mixture of surfactant and co-surfactant to achieve our desirable, suppose globule size, particle size or something. We can use combination of surfactant and co-surfactant, but it should be within 60%. Second point is when a energy formulation goes from stomach to the intestine, it has not got anything with the lipid part. But if the drug is having some kind of different solubility in acidic medium and alkaline medium, that may show different variations. But the lipids that are used, they basically start their work once they go to the intestine. They go to the intestine, they stimulate the gallbladder, it starts releasing bile salts, biles and bile salts, then micellar solubilization takes place. So lipid components do not have any effect whether there is a change in the or change because of the change or because of the gradient pH of GI tract. But if the drug is having some variation in solubility with respect to pH, that may behave differently. But the lipid part is not getting affected. So I would like to thank everyone, especially the entire team of Royal College of Pharmacy, for giving me this opportunity.
So it's time. No more questions just for my as a student. You know I always derive for some simple things, and I just want to note here the way he had presented and delivered. Everybody observed. He was very soft spoken and to the point. So he has cleared the doubts. And so many doubts in him that that's his expertise in his research. And he wants to make aware all the budding faculty members. So just I want to know about your experience. Regarding what are the challenges you face with these kind of motivation problems. That's my personal opinion.
Actually, I have shown only the success part. There are a lot of failures.
That's interesting.
When we started preparing SLN and NAAC, we got particle size like 1500 nanometer, 1400 nanometer, and we tried a lot of permutations and combinations. Like we got, suppose it has come to 800, 850, then also we tried to console that. In the definition, it is given 1000 nanometer, so we got a SLN. Slowly, we made changes. The major difficulty is if you increase the lipid component, it helps in controlling the release. But as moment you increase the lipid component, the particle size problem comes. It is increasing. So we have to keep it within a limit. Second major problem is it is not a suitable approach for high dose drugs. That is a major problem. That's for everyone. Because when we make suppose 30 mg, we have formulated cinacalcid hydrochloride with lipids surfactant. It is coming around 2 ml. 2 ml of liquid SLN formulation. Then we lyophilized it. So when we lyophilized it, it is found to be more than 1000 milligram. So making a tablet was a difficulty. So a lot of improvements are still needed so that we can. If you are going for polymeric nanoparticles, the chances of getting lower particle size is more. Okay.
Thank you, sir. Now it's time to recognize his expertise and involvement in the pharmaceutical sciences and development. So for participating our resource person, I would like to introduce our principal madam, Vice Principal sir, Professor Anil Kumar, Professor Vishnu Gani sir, Professor Kasturaj Kaveri sir, and Professor Madhusri Khandai, along with Professor Rajeshri Pandey madam. All the CPS alumni is particularly and partial here. All the CPS members, as I was our CPS student, and all the CPS members, please hand over our token of love, appreciation, and token of honor to Professor Professor, and request all participant faculty members to be last round applaud to recognize dedication towards research.
Manik bhai, hello. Am I
Hello. Hello. Yeah, I'm audible. Your audio is breaking. Am I audible?
Your voice is audible. Am I audible to you?
Yeah, you are audible. Right.
Namaskar.
Namaskar.
So we'll start your session now. So to introduce the next resource person of this scientific session, Dr. Manik Gupta from Birla Institute of Technology, BIT Mesra Ranchi. I'd like to request our faculty person, Dr. K. Harikrishna ji, to please introduce our resource person, Dr. Manik sir. Please. Then after we start the session.
Sure.
Good morning, one and all. My name is Dr. Hari Krishna, working as an assistant professor in Royal College of Pharmacy and Health Sciences. Today, I take the pleasure to introduce an eminent person, Dr. Manik Ghosh, who is right now working as an assistant professor in the Department of Pharmaceutical Sciences, Technology, Birla Institute of Technology, Mesra Ranchi. With more than 22 years of experience in teaching and research, his research interests include computer design, molecular docking, phytochemical analysis, and artificial intelligence and machine learning. Applications in drug discovery. He has published over 45 plus research papers in that field, Web of Science, and actively supervises research scholars in molecular modeling and natural product-based drug discovery. His expertise includes Python programming and industry-standard computational tools such as Autodoc and Glide. His research integrates traditional pharmaceutical sciences with advanced computational methodologies to accelerate lead identification and optimization from natural products. He is also the recipient of 33 intellectual property rights comprising 20 UK design patents, 1 German utility patent, 11 Indian drug design patents, and 1 Indian product patent. Good morning, sir. I would like to pass over the session.
Thank you. Okay. Can I share my PPT right now?
So I request Professor U.S. Mishra sir to remain as a chairperson for this session.
Device not connect. Sir, presentation है.
Yeah, sir, yeah, can you please share your presentation?
Yeah, just sharing.
Are you trying my way? Say computer skill expert look. Computer skill expert look.
Yes, sir.
Oh.
It is uploading right now. Just a minute.
It is showing the device is not connected.
The screen connected. Connected. This share screen connected. Sir.
Yeah, it has
Can you-
uploaded.
You are trying. Okay, it is coming again. No. Yes, yes.
Okay. I can annotate here.
Yes,
It
now
is-
the screen is removed.
Okay, but I can't try and take anything here. No? That is a problem.
Yes, you are visible and audible. Both your presentation PPT and you. Thank you.
Okay.
You can start, please.
Okay. Thank you all. Thank you for inviting me for this FDP program. Now, I'm as already the introduction has been done. I am working in this particular field, you can say, from last 22 plus years. Okay. As a faculty, I also worked in RCPHS for almost 2 years, and then I joined BIT Mishra, and from there I'm still in BIT Mishra. I work in the field of computational chemistry and natural product chemistry mostly, and my mostly the targets are antiviral and anti-cancer components. Am I audible? It is okay.
Yes, sir.
Okay.
Yes, sir. Audible.
Yeah. So today's topic is related with AI in pharmacy. We are going to discuss 2-3 different aspects of how we can use this particular system in our profession, along with what is actual AI and how they are using in the computer system architecture and how we can translate that thing in our system. It is a normal overview that has been given. So let us start with this thing. It is a concept that from prompt to drug architecture, the future of the pharmacy education, how we can do that particular thing, how we can start it from prompt and we can go into the higher state of that particular thing. Now, all of you know prompting because we know about Chat GPT, we know about the different type of Claude, etc. Simply, we are just typing something and it is giving us the output. So the thing is that the plain language program will be there. The design perfect drug, idiopathic pulmonary fibrosis, for example, concept has been given, and it is going to do something in the central AI, and it is going to give us a ready-to-use clinical output. Now that is prompt to the final drug. So right now, this particular insight is not there. Okay, so the key insight is that the paradigm shift should be there from the AI as a discrete calculator to the AI as a comprehensive project manager. So right now we are only using as you can say secretary. Whatever we want, we are going to give to that particular command, and they are just giving us the component. Now, next thing is that the hands-off charge in the traditional drug discovery, what we are used to do, it is the chem informatics, which we used to have it first, followed by the computational chemistry. We are going to do it in our lab. Then procurement of the component, etc. Generative AI, retrospective component is there, and then the final management, limb system, the assay management, etc. So the traditional siloed approach, siloed, you can say it is a word used for single lonely computer system which we are having right now. We are having a single system in which we are running our own programs. We are not you can say collaborating with the other companies or collaborating with the other systems for this particular approach of the AI or discovery of the drugs. So the traditional siloed system creates a collaborated together patchwork. Okay, cobbled together patchwork. Different teams are disjoined together. You can say this they are not joined, they are disjoined, but they are using different types of the tools for their particular work. For example, chem informatics is using their own tool, the computation chemistry using their own tools. They are not matched to each other. So we want a particular system that should match each and everything, and that should be present as a holistic system. The third point which we are going to do is the paradigm shift. The past system is that chem informatics, there is no, you can say, linkage in between computational chemistry and this one. The person is working on their own, they are generating some data, they are publishing it. We in the computational chemistry are doing the same sort of things, and we are publishing our data. There is no interconnection. But right now, the future which we want, and it is not a very far future. I can just suggest you, within 2-3 years, you will be getting this particular aspects. Okay, everything will be joined together. We have started joining it. Okay, everything will be joined together, and we are going to get a very good output out of it. Okay, there will be no discrete system. People will be able to know that what is happening in the cheminformatics and what is happening in the generative or retrospective system. They can utilize that thing in their computational chemistry, and they can work. And everything will be available with the help of the AI. And these things will be carried out by the AI. The human interface will be very minimum in each particular case. So why we are talking about this thing? Because it is the central orchestra sort of thing. A single system is going to work, or it is going to work as an agent, and it is going to work on different types of agent as a component, you can say. So it is a drafting thread, we can say, direct system of system approach. A central advanced reasoning AI is going to orchestrate that particular, you can say, collaboration of these individual system, or a silo system, and then it is going to have a complete background of this thing. So initial, if we go to the history and what we are doing right now, how the AI has evolved. So the first thing is the core capabilities. So initially in the traditional machine, which were you can say past 2014, only they are used to handling the noisy data or biological classification. The GPU power was also very less at that particular time, you can say. Applications, the general screening, target, druggable, etc. We are just going to find out that particular thing, whether it is targeted or not, druggable or not. For example, the simple docking studies, you can say. Then the output is interpretable target predictions only. Okay, but it was a very discrete sort of thing until unless we move to 14 plus molecular dynamics simulations. The molecular dynamics is actually going to tell us whether the docking system which has been carried out, means the molecule, if it is acting on a particular receptor, whether it is going to stick to the receptor in the system or not, because it is going to have a dynamic component. Here the GPU power has increased, and here we can predict. Okay, here we can predict the binding affinities. Now, post 2017, you can say the generative AI has already started coming. We were not aware of that particular thing, but still the companies, etc., they were using it. For example, the BioGPT, the Smile notations, etc., which still we are using in all of the PubChem, etc., or you can say ADMAT studies. And then the novel 3D molecule generation, etc., has started. And post 2022, you can say it is a multi-agent research. We are collaborating each and every component. It is not a solo intern system right now. It is a broad directive system, multi-agent system. So the future is not going to like a normal thing which we are going to use in our computer lab only. We will be, you can say, we will be mixing with the world. A person working in the Japan or a person working in America is going to have the data generated by our lab, and we can also utilize that particular data in our lab. So that particular standard components are very important. Okay, so the drafting thread is that we are going to have an expert from the biological database retrieval division, the expert internal or external physics-based tool, and then the dynamic experimental planning, etc. So everything is going to be controlled by the CEO or Central Advanced Reasoning AI, and everything will be taken care of by these particular capabilities. Now, the master blueprint, how we are going to go from a solo system to the multi-dimensional system, the API-driven infrastructure, if we are going to say. So the advanced reasoning LLM is going to have them. We are going to give the instructions. The instruction is going to be orchestrated. The evaluation is going to carry it out. The instruction is going to have, or it is going to have through a legacy system. Lab equipment like robotic automation will be there, humanoid in-loop physical system will be there, biological AI models are there, chemical AI models are there, and the medicinal AI models will be there. Now, right now, everything is present there. If you go and search the internet, you will be getting everything is just present there, but they are discrete. They are working in a solo system. They are not working as an orchestrated system. They are not connected to each other. Now, our system is not like that. We want the system should be connected to each other, and For example, why I'm telling this particular thing, right now you use a chat GPT system, simply type something about give me the biological activity of a particular drug. It is going to give you the biological activity. Now, again, the second question will be the what will be the chemistry aspect. Then you want to join those particular things, biology and chemistry. So these system, what it is going to do, it is going to take it from a particular portion or take it from a particular site and going to give you that particular hypothesis. For example, it is going to take it from the literature data, etc. So the biological discovery and target selections is a very important component. It is going to generate a hypothesis, and then the experimental validation will be carried out. So right now the components are there, but they are discrete. What we want to club them into a single system, so that you are going to type a system that I want a drug for these disease. It is going to do the literature survey itself. It is going to generate the hypothesis, and the experimental validation will be carried out, and within a particular time you are going to get the data. Initially, when we used to carry out the MD simulation, molecular dynamics, it used to take around 15 days. You can imagine, we used to have a GPU system, and we are going to keep our drug, and we have to wait for 15 days to get the data. And after 15 days, maybe the data was positive, negative, it doesn't matter. It is going to take the time. And after 15 days, we are going to know that the data is not suitable for the publication aspect than what we used to do. So it was a very difficult time at that. But right now, the same thing has been reduced to 1 day, 1.5 day. In certain system, they are doing in 2 hours or 3 hours if they are using the high graded computer HPC system. So you can imagine 15 days has been converted to 1 day or 1.5 day, like that. Now, so in future, we are going to get the data, etc., within a particular time only. Okay, so let us start with the node one, what we used to do in biological discovery and target selections. So the AI system is going to use that right now, like DORA system and omics system. They used to scan the article, multi-omic databases and biological databases, and going to generate computational data for you. Then based upon those particular data, we are going to do a gap analysis, and from the gap analysis, we are going to generate our hypothesis, how we can work on a particular target. And that it is going to be validated through a you can say experimental validation system. We are not going to do the experiment in our lab, but it will be carried out in the system only. So the generative AI pathway is the symbolic reasoning. For example, we are going to give a text prompt. It is going to convert it into a structure. Then the second pathway is the physics-based grounding, that we are going to do a docking studies, molecular dynamic studies, simulations with the help of the quantum mechanics, and everything will be orchestrated, and it is going to give us what type of molecule or outcome we are going to have, what will be the ADMET profile of those particular drugs. Everything will be given. Now, third point is your clinical prediction. And let us just quickly just try to understand. The first point is the biological discovery target selection with the literature, hypothesis generation, and experimental validation. Second point is the generative chemistry and molecular design, which we are going to do it. It is the second aspect. And the third aspect is the clinical prediction, because we want to predict that particular thing. We have generated it. We have designed it based upon the hypothesis. But whether it is going to work on the real world alignment or not, so that is also very important. So the capabilities of the system, the validation hurdles, and the feedback loop, everything is very important. For example, the model in silico, for example, they are prospectively identifying the patient populations, and similar trial design most likely to be succeeded. So already we are finding out which patient or which type of subject we are going to take for a particular clinical trial. They are going to track down thousands of the data, and from there they are selecting the patients. The validation hurdles. The AI system has not replaced the gold standard progressive clinical trial. Regulators remain the authoritative gatekeepers. Within the regulatory guidelines only, everything is being carried out. And the final thing is the feedback system. The phase 4 trial, which is a very critical trial, can be carried out along with the normal phase 4 trial with the clinical system, with the AI prediction system also. So the framework is your DMTA, that is design, make, test, and analyze framework that we are predicting that simply you give a design prompt to a drug. It is going through an advanced LLM system. The terms of the agent, teams of the agents will be there, and these particular teams of the agents are going to work on different aspects: biology, chemistry, clinical, and the real world evidences, and they are going to work together. They are not a discrete system. It is not a biology system which is not going to interface with the clinical system. Here, the biological system is going to interface with the clinical. It is going to generate the data. Based upon the clinical data, the chemistry can be changed. Then again, the clinical studies can be carried out, and everything is going to be checked in the real world with the help of your clinical data analysis, or you can say clinical studies. Okay. Now, this particular system is not a future sort of system, you can say. Still, it is working, and still many of the companies like Pfizer and Glaxo, they are using it. It is not so that it is a future system. It is present there, and they are using it right now. But a very important point which we used to face in our systems also, it is hallucinations. Whenever you are going to give a system a command and going to tell that you work on a particular aspect, the system gets hallucinated. It is going to generate certain data which are not true, and they are going to present it as it is. Because I was working in a particular project, I was just going through certain, you can say, this literatures. So I created a particular article and just given the AI system that you just find out what are the different types of the citation that can be possible for this particular article. Okay, I have written a 2-page article and just given it. I want to check that what are the different types of, whether the concept is true or not. So it has generated a list of citations that are matching with my article. So I was very happy that it is very good, no problem. The problem was that some of the articles were generated by the system only. They were not available anywhere. They were not available anywhere in any text, but it is writing, for example, ACS, Journal of Medicinal Chemistry sort of thing. It is just writing, and some XYZ has written this particular article. The name of the article is there given along with the DOI system also. So when I searched it, I was not able to find that particular article. So these things are known as the hallucinations. So obviously it can be done in any of the stages, whatever the 4 stages we have just carried out, it can be carried out at that particular stages also. So we as a human person, we as a scientist, we have to take care of these particular aspects. The AI is not 100% they are foolproof. So every time the watch should be there. So the hallucination is a fabricated citation, synthetic roots, and topological surface, etc. They are giving wrong data. I was generating certain images through the AI. I have given a data. For example, I want to generate a figure for publication. So I have given a data. It is an Excel. It has generated a very good data, very good picture. But the data which were present in the Excel sheet were not matching with the data which was present in the system, means the picture, you can say. So these type of things are also happening. It is generating itself. Sometimes it is hallucinating and it is generating itself. So that particular thing we have to take care of. We have to understand, and then we have to change. But we can use AI for n number of things. I can show you one example. I have just created it. At the last, I will show you one thing. Now, safeguarding the architecture, that's why we are just talking about integration validation. Integrated validation is very important. Confidence propagation, because allowing downstreaming modules to adjust the behavior if the upstream data has low confidence. So we should understand it, and we should manipulate the data. Not like manipulation, doing changes in the data. We should understand that what data is good, what data is bad, and we can change the data depending upon what type of system we want, the ultimate system, because the hallucination can often at the confidence propagation only it happens. Then the real-time adaptation will be there, and the human checkpoint. I will not say that the last point is the human checkpoint. Starting from the first integration validation to the confidence and the real-time adaptation, everywhere the human interface should be there. You can initially, I am not telling that maybe in the future they are going to be self-sustained, they can do it. But initially, when we are generating it, every time the checkpoint should be there. It is also a rule of our quality assurance. Now, the different types of systems which are also present there, physical bridge, for example, Himmler, Lupin, in case of the problem solving. Traditional legacy equipment, for example, if you want to have a pipette, microfluidic sort of thing, we are having this type of humanoid system also. They have been created and they are being scheduled by many of the companies who are working in this thing, because we know that the pipetting is a very important task for any type of analytical work, and the human error is a very important error which is happening. So they are just removing the human and they are giving the work to the humanoids. Now the educational imperative, why we are, because we are from the educational system only. So from the single system, if we want to go into the multivariate system or interconnected system, we should go in for, because right now what we are doing, the chemistry department is doing its own work, which is completely different from the biological department. We are not, we can say we are not working together. Okay, so this is a problem which we are facing. Okay, it is an isolated knowledge we are having. We are rigid in our disciplinary binaries. We are not thinking about what the other person is doing and how we can collaborate with that person and we can change. Second is the fractured workflow. At the last, you are going to get, the clinical department is going to get, because it is not getting the aspect of the chemistry which should go through the biology to give the clinical department aspects. So these things are now past, you can say, and we are slowly moving to the system where each and every department are correlated. If something is happening in the biological department, the chemistry is going to know, it is going to change the molecule accordingly, again going to give the biological department. Something is happening in the clinical department, similarly it is going to tell the chemistry or the other workflow department. So it is a unified network, you can say, and everything is happening in an orchestrated manner, and the AI is taking care of these things, because the AI system is in between, or you can say it is just taking care of what the component is going to have. Next is the translation, the blueprint, how we are going to have this. So traditional method is you're memorizing, synthesizing the pathway and drawing the 2D structure, generative chemistry. Target discovery where Silhouette molecular biology lectures, means single site only. And then the reviewing the past trial phases in the case. But in the AI era, everything has changed. It is the prompt engineering. We should know how to give the prompt. It is a very important thing. A same AI system can give you different result depending upon what type of prompt you are going to give to the system. So we should understand that thing in a very important manner. So prompt engineering for molecular design and critically evaluating the synthetic accessibility. Second is the cross-disciplinary omic integration, teaching a student to validate AI generated hypothesis against the literature. That is very important. They should not rely 100% on the AI system. Right now, if I'm going to give a student any project or any you can say assignment, I'm getting 90% of the students are using the AI and they are not changing any text and they are submitting like that one only. So it is not the problem of using the AI. It is the problem which I was talking about, the hallucinations. The AI system is going to hallucinate. We should validate those particular data with the normal system. We should understand the data. And again, the thing, AI generation, many of the you can say the manuscript, they are rejected due to these things. The book chapters are being rejected due to this one only, because they contain a high amount of AI generated data. So we should understand, and we should, based upon our literature which are already present, we should give the data. So whenever I used to do certain work or literature survey through the AI, every time I used to tell them that you please give a proper citation along with what you are writing. So that's why the prompt engineering is very important, how you are writing the prompt. The citation is very important, along with the data that is given generated by the things. And again, you have to check the citations also. As I told you, they are also hallucinating with the citations. Now, simulating trial parameter with AI model, understanding the AIMs and real world evidence of the feedback loop. It is very important for the clinical trial aspects. So the cultivating human in loop mindset is very important. Prompt architecturing, it is crafting precise multi-layer constant for the scientific queries. Then algorithm skepticism, for example, developing the biochemical intuition required for the spot hallucination, because we as a scientist, we know that where the AI can hallucinate and what point we should take in care for that particular thing. We should avoid that one. Synthetic infeasibilities, we know that this thing is not possible synthetically, but the AI is predicting it and AI is going to tell us we can go for it also, like that. And workflow orchestration, again, we should understand that which AI system we are going to use. Initially, I used to use Chat GPT for most of the work which I was doing. Then I switched to Claude AI. Why? Because I came to know that many of the things given by the Chat GPT, 80% is hallucinated. Right now, I don't know because I am not using ChatGPT. I completely switched over to the Claude AI. So why? Because I was getting a scientifically genuine result. I was validating it through many of the literatures, etc., and I was getting it like that one. So it changes. It is you can say the time is changing and the system is also changing. Now the workflow orchestration, which we are going to talk about. So it is the understanding how API-driven tools it is going to act with the design, make, test, and analyze cycle rather than isolating single system. So someone else is designing, someone else is making, someone else is testing, and analysis is also done by someone else. And one person is going to work on that particular aspect. Now it is not there. If making is being done, it should be based upon the design. Something is wrong, then the maker is going to tell the design person, he is going to change, then the testing will be done, analysis will be done, it is again given to the test and the makers, etc., and then they are going to give that particular molecule, and it is faster. If you go through, it is very, very faster. But very important aspects, the governance, ethics, and the academic integrity, because we as a scientist working in academic things, the governance and the regulatory aspects are also to be taken care of along with the ethical aspects of the AI use. So what is the interpretation? The teaching student at the LMNs are the black boxes, etc. Regulatory body legally mandate explainability for the tasks like mechanism of action prediction. Right now, we are not going for this mechanism of action prediction or SAR. Okay, if you want, I've checked the AI system also. Right now, another AI system is there, which is known as GPI. They are claiming that they are very good in chemistry. So I checked that particular AI system. I have checked every AI system for the chemistry, and for me, most of the system are failing in this particular aspect. I have given that particular system, write down the SAR or tetracycline and provide me the structure of the tetracycline and tell me which particular point are responsible. If I am going to change the structure, which particular point will be responsible, which particular number of the carbon will be responsible for that particular changes. So every AI system has given a completely different structure of the tetracycline, and none of them were able to identify the exact position in the structure. So this is the problem which we are facing. So there is a very, very high need of orchestration in chemistry system. Not for the other system, biology is there. The Sotex, they don't require those particular thing because everything is computational based, statistical based. Our thing is not like that. So the chemistry required a very, very high degree of precision in these aspects. We can't rely 100% on the AI system. Data privacy and very important, because AI tools normally they are free and they are taking your data, they are giving that particular data for the training system, and they can utilize those particular data for the other components also. So the legally it is very challenging, and we should understand that thing. And reproducibility, requiring a student to provide machine readable records, software versions, input data, and reasoning steps for the AI assisted system. In our system, in B farm and M farm, next session we are going to introduce the new syllabus, the PCI syllabus, which are having maximum of the AI system included in each and every semester. So if the students are not able to understand it, then it will be very, very different. And again, for these students are going to pass in a competitive fourth year. After 4 years, they will be having knowledge of the AI. Something I am not going to tell that 100% of the knowledge, but I can assure that for the day, because I have gone through the syllabus, if they are going through that particular syllabus completely, at least they are going to have 80% of the knowledge of the AI system, which are going to have. And if are learning the along with that, obviously for the pharma, it will be a very, very good thing. But what about the persons who have joined last year? For the 3 years, they will be not learning anything of the AI. Try to understand it. And they are going to compete with their juniors just after 1 year. The junior will be having all of the data with the AI system and the concept, everything, where to use, how to use, and everything. and just the senior, the batch which has joined this year, 25, 26, what they are going to do. 26, 27 batch will have an idea of AI. The previous batch is not going to have. We should think about these students also. There should be certain bridge courses. There should be certain things that they should have. Not only for the 26, 27 batch, means 25, 26 batch. It should be also for 24, 25 batch also. And the previous one also, who are in our final year or third year. We should think something for them and we should have an idea. There should be certain bridge courses, as per my suggestions. Because the scenario is going to change completely. If we are not aware of the AI, we are the person who is going to be fooled in the industries. It is not the system it is going to. System is already, it is advanced. They are working on those particular things. So the architecture of the academic laboratory is also important. Right now, we are not going to do it, obviously, but in future we can do it. At least not the laboratory, the classroom system should be AI enabled. We should make the student understand. I'm not depriving my student to use AI in the lab, but they should tell me what prompt they are using, how they are generating the data. It is very important because tomorrow they are going to work on these particular lab, these areas, and they are going to ask them, and they are going to get the 100% result out of them. So the API first design, treat every piece of the lab equipment as the API accessible module that can be eventually controlled by a central orchestration. Obviously, it can be done. Publish at every stage. AI foundation model need data. Publish target discovery, molecular design, and clinical insight broadly and immediately to train next generation biological data. Then program specific fine tune. Train multimodal data models uniquely for a specific equipment program, filtering out irrelevant component, for example, tailoring, etc., which require the things. Okay, so toward pharmaceutical intelligence, the AI agent, the researcher, the educator, everyone has to go through that particular thing. Prompt to drug is not a normal thing. It is coming, and most of the industries are using it. Now I'm moving to the second part of the thing, that is the connected healthcare blueprint, which we are talking about. Even the FIP has started a strategic synthesis of FIP global policy and our PCA education guideline 26. So we should go through and we should understand what they are going to ask about it. The traditional tech was building about the capabilities, static, okay, data storage and manual retrieving was there. But the predictive AI is going to do the identify non-adherence and flagging the adverse drug reaction, etc. But right now we are moving to the generative AI, the frontiers, etc. The novel drug design, repurposing of the older drugs, etc. Generation of the personalized present education, everything is being done by it. And if the students are not knowing it, obviously they are going to fail in the world tomorrow. AI domain capabilities are high-speed data entry, administrative automation, massive real-world data analysis, complex inventory robotics, and the cross-referencing vast drug interactions. Human domains are what? Contextual clinical judgment. We are going to do the clinical side only, the empathy, empathetical aspect, which the robots are not going to do, the AI system is not going to do. But I have seen it is not so. If you are going to ask certain question in a particular manner, the AI is also going to do something in the empathetic manner. They are getting more empathetic than a person you are talking to. I have seen in many of the cases, you simply go to the chat GPT and talk to it and ask any question in a rude manner. Okay, and ask any question in a very polite manner. You will be getting how empathetically it is going to give the answers. Okay, ethical overview and accountability, and then validating algorithm and truth. So the AI has a potential to descriptive repetitive task. Okay, but creating opportunity for pharmacists to assume more of this particular thing, the specialized thing, is very important. So we should, as a human, are going to do this particular one. Now the imperative for the human oversight, what the thing is that the raw data, medical databases, etc. What happening right now is that, try to understand, a patient is going to a particular doctor, we are checking it, a drug is given, he is not getting, you can say, relieved by that particular drug because he may or not have taken the proper doses or at that particular time, or the complete dose has not been completed. But what he or she is going to do, he or she is going to switch over to the another doctor. Now the another doctor is going to start with a new drug or something like that, and again something is going to happen to the patient. We do not have the background of the patient, what he or she has taken earlier and what we are going to do right now. Okay, so this thing is very important thing. The AI algorithm works. It is going to tell each and everything. It is going to tell us. It is going to have a verification checkpoint of pharmacists for safe and verified patient care. Okay, so if the data are being stored, they are being utilized in a proper manner, each and every one us will be in a safer way. Okay, so elevating the professional ecosystem, you can say workforce redesign from the administrative burden to the clinical and constructive consultative capacities. Okay, so we should go for that particular thing. We should be proactive intervention, personalized medicine, and patient engagement is very important in near future. So the strategic ecosystem playbook is the government policymaker. They are going to do the regulatory aspect. We as an educator and things, we are going to make the competency and curricular aspect for the future generator pharmacist. Patient and advocacy, the literacy of the patient is very important. Okay, why the literacy of the patient? I've seen, I'm living in an area which is very prone to malaria. Initially, it was very prone to. Right now, the thing has subsided. But when I used to go to any pharmacy and going to tell them a person is suffering from fever, they used to cut two chloroquinol and 2 tablets of paracetamol and used to give to that particular patient. Okay, now try to understand whether the patient is going to be revived by only 2 doses of chloroquine, etc. Whatever the drug they are, pharmaquine, chloroquine, whatever they are taking. So only 2 doses. Why? Because the patient is not very educated, or the patient is from a very low background sort of thing. Okay, so this thing is very also important. Right now, it is not. So everyone is having AI. Everyone is having many of the components. Okay, so the pharmaceutical industry validation is also important. Similarly, the practicing pharmaceutical or pharmacist clinical application is also important. So the first pillar is the government or the policymaker, which already they have done the infrastructure, etc. They have created. They are making the regulations and they are working on the ethical aspect of the AI. Okay. Second is the pharmaceutical industry and the scientist, because we are generating that particular thing. We are generating the scientists from our academia. So the phase one is the drug discovery trials. Our students should understand these things. Data integrity is very important. How they are going to do with the data, how they are going to learn with the particular data, and the clinical validation of the data with AI accountability. What we used to do is the foundation. The main thing is the foundation for both of the thing. The previous what we have gone through the industrial aspect, the foundation is very important. That's why the courses have been changed. Adaptive learning is very important. Virtual simulation is important. Lifelong scaling is very important. Okay, so adaptive learning, AI driven system delivering personalized learning experience and identifying knowledge gap in the real time. Normally it is not possible in all of the colleges, etc. At least we as a teachers, we should understand these things and we can change the student aspect through the AI aspect. The immersive realistic patient scenario to build clinical decision making, real world deployment, how AI could do that particular thing. And lifelong upskilling is a very important aspect. So the blueprint of the action that PCS labels has given as per the NAP is that pharma knowledge, analytical, etc., ethical integrity, everything will be there. But these three aspects, that the patient-centered care, digital technology awareness, and innovation and problem-solving evidence of the solution, it is very critical. And all these three things are based upon the AI. You can say we can use the AI in these particular aspects, and we can change the scenario. Our students are going to work well on these particular aspects. So fourth pillar is the practicing of clinical pharmacist, which normally we do not have in India. Okay, because the doctor is prescribing, he is giving the drugs. We are not going to tell anything to the patient. We are just giving the drug to the patient, is taking it, and we are not going for that particular thing. But right now it is going to change. The pharmacy aspect is also going to change. Predictive analytics is very important. As a pharmacist, we should, the person who is working in a clinical aspect, they should take the data, they should keep the data, they should analyze the data. For what? For the precision medicine, etc., or for the clinical decision support. Okay, it is very important, because right now we are not doing these things. As a pharmacist, we are not doing this thing. Maybe because of the rules, regulations, we are not able to do it. But if you in 1 or 2 days, if you remember, before 1 or 2 days, this IPA, BCI, they are just telling CDCS is also telling that AMR disclosure should be kept at each and every pharmacy. You should have a QR code. The patient should update any type of the adverse drug reaction, ADR, etc., to that particular thing. So this thing is enhancing, and slowly we are moving towards this clinical decision supports. Fifth pillar is the patient and the advocacy, because the patient is the main person. Anything in the keyway also, we know that if something is going to happen to the patient, then the drug is not going to work. Okay, so the education, the discrimination of this particular role, the AI, etc., to the patient is important. The transparency, the advocacy of the clear communication is very important. How we are going to, and that thing is going to be influenced by the AI. The trust of the patient to the pharmacist, trust of the patient to the AI system, etc., is also going to be verified. And the equity, who is going to share? If something is happening, equity is not only for the profit, it is also for something is happening to the bad, who is going to share that particular thing. Addressing digital literacy and cultural diversity to ensure no patient is excluded due to inability or engagement with the digital tools, etc. So these are the optimal patient care thing. Already we have discussed it. Okay, so this is the horizon, the new roadmap. So oversight of the digital therapeutics. Okay, it is regulated AI enabling device and generative AI health application. Pharmacists will bridge the gap between AI innovation and clinical patient, requiring specialized knowledge AI system, data integrity, and workflow. So I'm not not gone through each and every aspect of the AI that is giving in the syllabus, but if you go through the overall thing, it is going to give you a very broader aspect of the pharmacist. So the 3 point, the FI commitment that has been given is that AI should serve to enhance, not replace the clinical aspect for all this particular year. Then AI, the clinical judgment, processing and integration of the pharmacist, and AI should serve to enhance, not furnish the personal interaction of the pharmacist. So it is going to help. Okay, it is just going to help. So AI should serve as the enhance, not replace, and the clinical judgment and the personal interaction of the pharmacist will all be there. Okay, so thank you all for your patient hearing. If you have any query, anything, please.
Thank you, sir, for presenting such a nice topic, and which is relevant to the our Pharmacy Council of India recognized FDP program team. And this the topic which that is computer application of AI in pharmacy, which is really a useful topic to all of us, and how you utilize the AI in research field. And as today morning, actually I asked to one of my teacher colleague about this AI, because we are the old generation. We have very less idea about this AI, but and I asked him actually about one of the handwritten slip present in my table actually. That time I asked him actually how actually I can use the AI tool, and he saw me in ChatGPT already. He that took the photo and he placed in that ChatGPT. I think actually I have very less idea about that, and he converted that one to the Word file.
Yeah.
So from that time onwards, I can able to understand what is the importance of AI, and how that will be helpful, and which will be more helpful to the young generation. They can capture all these things in a very fraction of time. But as per the today's requirement for all of us, this AI, which will be the more important to all of us to save our time, and which having lot of application in the so many fields. And you highlighted that one, and which will be helpful to all of us to solve our problem, and what are the different tools of AI, and the different tools how will apply in the different segments, all these things. And really it's a very wonderful session, and you spent your valuable time to explain or to explain in more detail in a very simple manner to all of us. And it's now time for questionnaire. So,
Dr. Manigus, namaskar. Dada.
Namaskar, namaskar, sir.
This side, Prashant Chaudhary.
Yeah.
I am with wow, we are from the beginning. Saraj already told old generation and mid generation,
Yeah.
and the Gen Z is there. So my one question is particularly simple. Though I started using these AI tools, just can you help us out how to go for better prompt writing, which is the best and easiest tool for the beginners to know about prompt writing?
Prompt
Which,
writing.
yeah,
No, the main important thing of prompt writing is that who is going to write that particular prompt. Try to understand. Okay, I as a teacher, okay, if I am going to write something about it, I want to know the answer should be written as a teacher. Okay, so you should make the system understand that who is writing that particular prompt and for what he is writing. For example, I'm going to tell that act as a professor. Okay, so the AI system will understand that it has to act as a professor. If I'm going to write as an act as a +2 student, or tell me this particular thing as a +2 student, or let me understand this particular tool as a layman. Okay, so it is going to understand that for whom we are talking about and who is talking about. Try to understand these things. These are very important aspects. So if I'm going to talk something about a particular research paper and just given it a research paper and going to tell me something about it, or just go and just let me summarize this particular thing. So it is going to summarize like itself. But just tell me if I have uploaded a paper of pharmacology and going to tell it that act as a professor of pharmacology and summarize this paper based upon these particular aspects, because I know that what aspect I want from it. You will be just getting the changes. You do it today. I will just assure you, you will be getting the changes. So these prompt because, and again important thing is that the second thing is that the AI is going to tell you how to use the prompt. You just give that particular thing to the AI and ask that if a professor is going to ask a question, what should be the particular prompt for it? And what are the important points a professor should include in this particular prompt? So AI is going to generate it. You just go through it, try to understand whether you want it or not, whether you want to delete something. You just take that particular thing, you can go for it. So the AI prompting is very, very important thing, and we should understand it very critically, because the same system can give you n number of answers. The hallucination will be also less in these particular aspects, because it will be understanding that certain specified person is asking. I have uploaded my profile, okay, to the AI system, and just tell me in the Claude AI, it is just giving that whether you want to upload the background, etc. or not. So I've uploaded my background. Now that particular system is giving answer based upon my backgrounds. Now I do not have to ask that particular thing. If I have also given access to all of the other chats which I have made throughout. So it understand that what is my work criteria, how I working, how I am working on a particular aspect like that. So whenever I am asking certain question, it is again asking question whether you want to go for this particular aspect or other aspect. This thing also, I have given the option to the AI that please ask me before doing something. I have given a command, it is generating the text and it is giving the text to me in Word format. Okay, so the thing has been given to it that you give that thing in the Word format. If you allow me, I can show you one thing. I'm sharing my screen. I can show you one, two things.
yes, sure.
So another system has been. I'm just removing it from the studio. My system has been shared. Are you getting it? No. The whole system
yes.
I shared it.
Full
I'm
screen.
not able to. No, slide. Please remove from the studio. Okay. I have shared my screen. Are you able to see it?
Okay.
Yeah. This is the screen. I can show you one thing. Let me minimize it. Uh, no, not minimize it. I can write it here. You can also go for it. Manik Ghosh. You just type
See,
simply
just,
3
sir, just let our participant to do the same. All can do the same in your browser also, mobile browser.
They are able to see it?
Yes,
I
yes,
think-
all can see. All participants are there, sir. We are just obstructing them, so we are going back to our seat and let them
Yeah.
see all the.
Okay. So just check it. So this is my profile, B.I.T. Mishra. You can go simply type Manik Ghosh B.I.T. Mishra. It will be coming. You go to this lab, okay, Arohan lab, which lab group which belongs to me. I've just created it. You will be getting many things. I'm not going to talk about many things about this particular lab. This is the research team which we are having, the research which we are doing, the research team which we are having. My PhD students, etc. You can collaborate with them also. They are in very good profile. For those who are passed, they are in very good profile. And these are the present students who are working. The main thing I want to just show you is this statistical advisor tool. Are you able to see it?
Yes, sir.
Yeah, here you can see Arohan Lab Statistical Test Advisor. Okay, so it is a tool that has been created by AI. Now, why I've created this particular tool? Because we, as a pharmacist, we as a student of a pharma sector, statistical analysis we have to do for each and everything. If you want to publish any paper or something like that one, the main problem is that if I'm having a data, whether I should go in for a T test, ANOVA, a chi-square test, or n number of tests are there, we are very confused. Okay, so for that particular thing, I just created it based upon my knowledge, based upon the thing, given the prompt, and it has created complete AI system, or you can say agent for it. Simply here, you just choose the file, for example, any of the file, and I can show you if I have stored it. Let us just create a data quickly. I'm just creating it. You will be able to understand it. Here you can upload the data or the paste the data also. Okay, so whatever the things you required, you can upload it or paste it, and simply it is going to do. That is the thing I just want to show you what AI is going to do. Okay, it is not only about getting the things done. I mean, asking the questions and getting the thing done. It is way beyond it. The whole system, it is a software. It is completely being generated by the AI. Now here we are having the system. Yeah. We are already. I've just downloaded this thing. We are having multiple regression, etc. It is written. Okay. I am downloading it. I think so. I've shared it here. AI agent tools. Okay, it is already shared there. Okay. So we are going for documents, and here it is AI agent statistical tool. So these are the tools. I will be giving it to you. Just go it. One way ANOVA. Are you able to say it? One way ANOVA. I'm just clicking it. I don't know what the system is going to give. So it has just checked the things, and it is going to say whether it is a normal data, ordinal data, etc. So it is a normal data, it is saying. Okay. Then there are two types of tests which we can do. It has already predicted this data. We are not going to predict. Already it has predicted. Now we are going for the analysis or recommendations. Now it has selected all of the groups, and it is going to predict or give you the recommendation based upon it. You can, if maybe it is on the same subject, we are doing certain studies before giving the drug and after giving the drug. So if that will be there, you can click it. Otherwise you just get the recommendation. Now here you can say one way. It has given a recommendation based upon certain decision pathway. It has made wide format, each column one group, placebo, drug, four parameters, independent group, placebo, drug A, drug B, normal. It has given the mean, it has given the standard deviations, variance ratio, everything. Based upon the previous calculation, it is suggesting us we can go in for one way ANOVA. It is also suggesting that if assumption does not hold, you can go for different types of analysis. You can say rich analysis, ANOVA, etc. it is also giving you certain codes, Python codes or R code to run on those particular softwares. And then you run the analysis. Now here you can see it has given us the complete data. What is the value of f, what is the value of degree of freedom, 336, p value, h values, etc. And then complete data analysis. Not only this one, it is going to compare placebo with drug A, placebo with drug B, everything, and going to tell which significant level it is there. And then also it is going to give you a whole writing. You do not have to write. Try to understand. You do not have to write anything. The data is being generated by the system only, and it is you can say APA style write up data for your thesis, for your article. And again, it is going to give a very important thing, the graph, if you want. So from here also you will be able to understand and see it. If something is wrong, you can go and do the analysis once again, and you will be able to understand. You can change the data, you can change the variance, etc., and you can just go for it. Okay. So new file, new data we want to take. For example, we can choose any of the. For example, I want to go in for two PRT test sort of things. Here it is given a PRT test. Okay. So before treatment, after treatment, which I was talking about. So just group analysis, before treatment, after treatment, same subject. Okay, recommendation. It is giving us a recommendation of the peer test. Now you can understand. So it is a system which is generated by the system. Okay, I just checked it in between. So we as a scientist, that is our case. It is giving everything. The write-up, etc. is given. The table has been given. The graph has been given. Simply you copy paste it and use it. So it is one of the examples. Just I have given you one of the example which in the AI system, what we can do with the AI system, what are the different components which we can do with the AI system. Okay, you can do n number of components with the help of the AI systems. So I will just request you just go and use this particular system once, okay, and you just check it, and I will also want your feedback on those particular aspects.
Sure, sir. Thank you for demonstrating all these statistical test tools. Thank you. We will be in touch, and we will be again getting back in touch
with you. I'm always with Royal.
Okay. Okay, thank you, Dr. Manik sir, for giving nice presentation about the AI, and you clarify our doubts in a very nice manner. So the same support also we expect from you in future also.
Sure, sir. 100%.
Okay. Thank you, sir. It's our great pleasure and privilege to listen from you. Okay.
So we are going to present a memento for you. For which we'll send it. We'll send it.
Thank you.
Through post. Okay. Thank you.
Okay, thank you.
Thank you, sir.
Thank you. Thank you.
Manik bhai, thank you so very much
Thank you.
for
Thank
accepting
you.
our invitation and giving your valuable time.
No problem. You can call me anytime. I'm always there for a while. There's no problem.
Thank you so very much. From the beginning, you are associated with our college. You are the pioneer staff to this RCPH family. Thank you
I'm
so
always
very
there.
much.
That's why I'm telling you, I'm always there. It is like my home. I'm not going to tell my second home. It is like my home only. Okay.
Thank you. Thank you so very much for your kindness.
Thank you. I'm logging out.
Thank you. Thank you. We'll meet in later future.
Sure.
Thank you. Take care. Have a nice day. It was a very nice presentation. Brilliant. Thank you. Thank you so very much. Next.
Now it's your.
Thank you all. So with this, all our scientific sessions we covered 14 online programs, 14 physical programs, scientific sessions, and 5 online programs. The support of all you were able to do it. And our next schedule is the Wednesday sessions. So we have little gap in between. So I like to request our Prasad sir and Rajesh just to do some cultural activities in between, just so that we all will relax for a while. Teachers were reading and listening, which is very tough and painful work only. So just relax for some time before the starting of this very activity session. So Rajesh sir and Prasad sir may read this. So start. They came up. Okay, so without any time, first we will complete this very detailed session. Our chairman and secretary are there reached. Then after we will do some cultural activities, which will be followed by lunch. Thank you.
those persons who have shared their knowledge, and I would like to all the delegates also for the presence, participation, and presence sharing. Now, before starting the valedictory, I would like to invite our dignitaries of the dais. First, I would like to invite the president of this FDP, Dr. T. Prakash Rao, sir, Chairman, Berhampur of the Pharmacy and Sciences. On to the dais. I request that for our guest, the floor. Next, I would like to request Shri Sagar Srikanth, sir, Secretary, Royal Education and Research Society, onto the dais. Request our faculty members to give our letter of welcome. Now, I would like to request our principal line, Professor Dr. N. Usharani, coordinator of this FDP, onto the dais. Next, I would like to request our Vice Principal, sir, Professor Dr. Umashankar Mishra, sir, onto the dais. Next, I request Professor Dr. Amrit Kumar Mahapatra, sir, Organizing Secretary for this Secretary Development Program. Now, I request Dr. T. Prakash Rao, sir, President of this Faculty Development Program, to give his remarks on this 1 week long journey. Good afternoon. Respected Secretary, Srikanth, our Principal, Madam Vice Principal, Ma'am Sir, and the Organizing Secretary, and Dr. My dear faculty members and the faculties from the other institutions, those who have had a nice time, nice academic feast, what I can say, which was most essential as we have thought of, because the changing trends and the changing needs of the students and changing presentations, changing modalities of teaching and learning, and different facets of the other skills and tools. We had a very good interaction with the eminent, distinguished specialists in this regard in different fields. I congratulate all our team members, our professors, our coordinator, madam. Especially I thank to Professor Nanjan, who have put maximum heartful efforts to bring these things to our forum and make it happen. And I feel that I am able to satisfy our management, satisfied with the performance of the total team, both teaching and non-teaching, as a teamwork, as a sportsman spirit, or academic spirit. They have put the things into happen. I will individually thank all the members of our college. They have done their job with precision and timely. That's most important. Time is most important. Every session has happened as per the schedules, and the outcomes, everybody had a good academic content. They have enriched themselves. At the end of the day, I feel that this type of CPE, continuous pharmacy education,
including the students is necessary because there is vast area of applications. Besides what the basic pharmacy a student learns here, that is not sufficient. But the changing world scenarios and changing needs of the industries and their counterparts are organizations. So we have to inculcate the many new things to the students through our faculty development program. So this is a opening from our side. We can have some more things in this regard in different areas of interest, particularly coordinating and patent research activities. Yesterday, when we had a very good session, we were also enriched with the ideas, but simply a small presentation or book publication can be patented. So enthusiastically, everybody has performed the outcome. Everybody might have seen it. ମୁଁ ପ୍ରଥମ ଦିନ କହିଥିଲି ଦେଖ ଶୁଣ ଶିଖ ଆଉ ଅଭ୍ୟାସ କର and put it to the academic things. This is a small prayas, small implementation where we have started the things, and this prayas should percolate downwards to the students. That is our utmost aim. We enrich ourselves and we make our students enrich in all facets and all. We have to make them aware that they can do things wonders after learning so many new things. Yesterday, some professor has told that only book reading is not sufficient. So many things have to be inculcated. These 4 years or 6 years stay of the students at least any institution should be more fruitful and more productive when they come out, and there will be more confidence building. This is most important. They can do that thing. They have to develop. You can be student who are meant be. That is most important. Confidence building. Trust on yourself. That we can be much more fruitful to the our academics and much more fruitful to the society. Thank you once again. The professors, particular and professors, the good efforts to make this thing to happen. Thank you once again.
Thank you, sir. Now I request our esteemed secretary and Srikanth sir to express to us.
Respected Chairman, sir, respected Principal Madam, Professor Tushar, respected Vice Principal, sir, our Professor Anjan Kumar Mahapato, sir, Organizing Secretary, and respected resource persons, and respected faculty members. A very good afternoon to everyone. As we come to the conclusion of this 1 week faculty program 2026, I feel a deep sense of satisfaction and pride. Over the last 6 days, from 6th July to 11th July, we gathered here our RCPHs with one common goal: to learn, to unlearn, and to relearn. the theme teaching pedagogy basics and advances in research and applications of AI in pharmacy. We had the privilege of learning from eminent resource persons who took us to topics ranging from flipped learning, simulation-based education, AI research, and other training and research funding, which each session gave us new perspectives on how we can make our classrooms more engaging and our research more impactful. On behalf of Royal Education and Research Society, I extend my sincere gratitude to our respected Principal Madam and Vice Principal Sir and Anjan Sir and Madhu Madam and other faculty members for their constant encouragement and the leadership. And I also thank our Honorable Directors team for their presence and support throughout this program. My heartfelt thanks to IPGA and BCI for their collaboration, to our Professor Anjan Kumar Mahapatra sir and the entire organizing committee for flawless coordination, and to all the resource persons who shared their knowledge so generously. To all the faculty and trainees, congratulations on completing this FDP. I hope you take back not just certificates, but ideas, techniques, and collaborations that will benefit your students and institutions. I request you to implement at least one new pedagogy or tool you are left here in your own classrooms. Let this not to be the end, but the beginning of a learning network among all of us. In closing, I once again thank everyone who made this FDP a successful one. May our Royal College of Pharmaceutical Health Sciences continue to be a center for academic excellence, research, and innovation. I also expect same support from all other institutions also, so we can make it in more colorful way. Thank you.
Thank you so much, sir, for your kind words. Now I request the coordinator for this FDP program and the principal, Dr. N. Usarani, to please address and express few words.
Good afternoon, all of you. Respected Chairman, sir, Dr. V. Prakash Rao, sir, respected Secretary RERS, Sri Srikant Chaka, sir, and respected Vice Principal of Royal College of Pharmacy, Dr. Umashankar Mishra, sir, and the respected Organizing Secretary for today's event, Dr. Anjan Kumar Mahapatra, sir. Today, I really thank for giving me this opportunity. I take the gratitude and pride to say that we have come to the conclusion of this faculty development program. In my opinion, the conclusion is only for today's program, but still whatever we have learned, the knowledge that we gained, the insights that we have seen, and the hands-on trainings that are given by certain resource persons, and some of the lectures on outcome-based education, and regarding patents, especially patents and publications and their impact, and how we have to conduct ourselves along with all these things. The objectives have been well designed. What all I have said now are all the objectives of our program, like pedagogy, then basics and advances in research, and implementation of AI in our daily academia. These were the main objectives of our faculty development program. I hope and thank Dr. Anjan Kumar Mahapatra, sir, who has rightly brought the legendary resource persons to the program, and the objectives have been well filled. And the second part is now the outcome, where how the outcome is, the expected outcome is satisfied or not. can only be known when all these things are carried to our classrooms, when all these research activities are carried on, or all these novel ideas and insights are carried on to our laboratories, to our classrooms, to our research areas, to our publications, and to our papers. Then the expected outcome of this faculty development program will be reached. And I hope that would definitely happen because the resource persons work in such a way giving us the knowledge. I also thank Anjit Kumar sir for having given me the opportunity to be one of the resource persons of this FDP program. I also thank our Professor Sujit sir for giving me hands-on training on certain of the instruments. Of course, Anjit sir also has given yesterday his hands-on training program was going on. On this occasion, I would thank all the faculty of Royal College of Pharmacy, the technicians and the office staff, everyone who have contributed to their level, who have moved out of their comfort zones and have made the event successful. And yesterday's lecture really, as Chairman sir has said, it was really impactful regarding patents and publications and what Sir Pawan Kumar sir has told yesterday. I was actually working on certain new method developments, which is my area of research, that is new analytical method developments and validations. I have recently developed, and then those are in the pipeline towards publications and patents. But yesterday, some of my doubts have been automatically cleared, where Sir was telling better go primarily for a patent and then for a publication, then we have 1+1 offer. And on this behalf and regarding this, I extend my hands towards our own in-house faculty as well as the faculty of the guest faculty who have reached to this program. You can all are free to join with me to collaborate in my research work, and I am free to collaborate with your research work. And let's move the work forward quick because two heads are better than one, so we can do better when we collaborate with each other. And also expect these type of programs in our neighboring institutions where our faculty will go and inculcate and collaborate with them, and so on. I expect these type of programs also in future. And I thank on this insight that for active participation of all the faculty, and thank you.
Thank you so much, madam, for your addressing to the gathering and nice words. Now I request our Vice Principal, Professor U.S. Mishra sir, to express the word.
Thank you, Dr. Ramzan. Respected Dr. Sri Prakash Rao sir, Chairman IPGA, respected Mr. J. Srikant sir, Secretary IPGA, respected Sridhar sir, respected Dr. N. Usarani, Madam, Principal, IPGA, respected Dr. Ramzan Kumar Mahapatra, Organizing Secretary of FDP, respected all my junior and senior teacher colleagues, respected all the non-teaching staffs present over here. It's my great pleasure to share a few words regarding the FDP, which was started since July to today. And during this 6-day FDP, we learned lot of things, and the research persons, they all are very well explained about their expertise and which will be more helpful to all of us because during this 6 days program we focus on the different segments like the research and academia and what are the how that the AI tools will be helpful in our research and day-to-day activities. So which will be more helpful to all of us. And during this 6 days program, we learn all of us a lot because we all are experts or we have idea about a specific field only. So but where in this activity we cover most of the field and which are unknown to all of us. So really this activity which is a very useful, informative and helpful to all of us. And today that is the last day of the activity which during this last 6 days this all the programs are covered successfully and meaningful, and this is only possible due to the teamwork by all the staff members of Royal College of Pharmacy and Health Sciences, and I extend heartfelt thanks to exclusively to our organizing secretary, Professor Ramjan Kumar Mahapatra, who guided all of the committee members and he struggled a lot to make this activity successful. And I extend also my heartfelt thanks to Madam Professor N. Usarani, Madam, who is the coordinator of this FDP. And lastly, I extend my heartfelt thanks to the participants, those who are coming from outside, from our means other college than our college. Due to their presence, we make this activity more successful. With these few words, I extend once more my heartfelt thanks to all of you. Thank you.
Thank you, sir, for those inspiring kind words. Now, I would like to request Professor Dr. Ramjan Kumar Mahapatra, sir, organizing secretary, to say a few words at the same time for the country.
Dignitaries on the dais, of the dais, all the teachers and friends. A very good afternoon, one and all. It's my customary to give a short report of this program, of this 1 week faculty development program that we had conducted. It's my privilege to express the contributions of our respected chairman sir, secretary sir, and other governing body members of this Royal College of Pharmacy and Health Sciences for the all-time support, and in particular for the support to to conduct this faculty development program for 6 days, which is officially recognized a 1 week program. Thank you to our management members for providing and making available this beautiful conference hall in a very short span of time. Definitely they took lot of responsibility, I can say, to arrange this hall before starting of this FDP program, which made everybody comfortable for all the 6 days to run this program. Thank you. The appreciation supposed to go to the IPGA Odisha State Branch for the association for this program, and financial support to what they declared 25,000 will provide for this program with this satisfaction without asking. For this, on their absence here, the governing body members and the family members of RCPLS would like to acknowledge them to the IPGA members, Dr. Lajit Kumar Sahu sir, Dr. Sudarshan Biswal sir, Dr. Biharanjan Padi sir, the IPGA members, and once again thanks for the presence in the first day inaugural ceremony of this FDP program recognized by the Pharmacy Council of India. Special thanks supposed to go with Dr. Sudarshan Biswal in arranging the resource person, persons, Sri Rajat Kumar Mahapatra, the president of Bharat Biotech, and the senior scientist, T. Pawan Kumar ji, and taking better responsibility in their arrangement and make them convenient to reach this destination and contribute to this program. And special appreciation to Dr. Biswal for his special responsibility and major responsibility to obtain the Pharmacy Council of India (PCI) New Delhi recognition for this 1 week faculty development program. Appreciation supposed to go to all the resource persons contributed for this 1 week program. There were 14 resource persons contributed physically and 5 in virtual mode or online mode. Starting from the first inaugural session, the contribution is the chief guest, Dr. Mahanti, the Vice Chancellor, Accredited University, as a resource person, to as a chief guest for the program. His contribution and presence definitely made the program colorful and successful. In the first day, the other 2 resource persons were Dr. Sudarshan Biswal, who had experienced some soft skills and teaching methodology, and Dr. Usarani Madam on teaching pedagogy and new teaching technologies. It's appreciable. The second day was, and the other resource persons, Professor Mike Udaramani, the controller of examinations, Odisha University of Health Sciences, OUSS, Bhubaneswar. We need to appreciate for the presence and traveling from Bhubaneswar to Berhampur. Rajat Kumar Mahapatra, who contributed his responsibility yesterday. Professor Kunamani Rena from RIT, Berhampur, the principal. Dr. Sagar Kumar Mishra from Utkal University. Professor Vishwaranjan Mahanti, Dean Research and Professor, Dreams University, Kandy, Cotton. Professor Alok Ranjan Biswal, who explained on IPGA and in teaching curriculum and basic requirements. Now it became basic requirements to meet the requirements of FDP or NAAC. He expressed well. Dr. P. Pawan Kumar yesterday who gave the talk on IPR. Professor P. Subodhi, Professor C. Niranjan Patra, and the coding, pharmaceutical and medical coding team. I would like to appreciate all and the Vitlam board who contributed, Professor G. P. Mahathasa, who explained some new national education policy, Dr. S. Srinivas Kandasar, Dr. Pritish Kumar Bharadwaj, Sri Dhananjay Panigrahi, Dr. Malik Kumar Bose, all on behalf of all the team of this FDP would like to express our thanks to the resource persons for their contribution. And special thanks and thank goes to Professor Sudeep Kumar Sahuta, our faculty member, one knowledge source, for his contribution with excellent training for most days, especially FTIR, DST, UB, HPLC, all the instruments. So I hope all might have gained a lot from the excellent training. Thank you, Sudeep sir. Appreciation on behalf of this organizing committee goes to all the participants, 31 from outside, from different institutions of Odisha and out of Odisha. Many attended from Jaipur, Baleswar, Sambalpur, Vijayanagaram, and other places. And from local colleges also. Thanks all for attending the program and participation. We had arranged their accommodation, those who come outside the hostel also, hope for a happy stay and peaceful stay and stay for the days they spend here. All the staff members, non-teaching and teaching staff, they supported a lot, worked hard even on Sunday. As organizing secretary, it is highly appreciated for all, and we are so happy for the contributions. Just overall, just little I will tell from the outcome. See, just 2 words and learning outcome from this activity. I will tell that is awareness, which is required to start, and follow, which is required to continue. These words sounds like definitely words are simple but having weight. And the chairperson and chairman of our college, being one eminent doctor, I'll give a small example. Suppose one excellent, very good surgeon did one surgery, but after that, the patient will not be properly followed. He or she may go to sick and so follow in every step is essential. And what we learn and what we gain, we need to continue with that. Suppose someone is giving farewell. On that day, lots of things we are experiencing, lots of praise, guidelines, appreciation, class, standing ovation, so many things we do. But after that, then occasionally, neglectfully, we do a further contact. So follow continuation of things. That is important. That is our personality and our social connectivity. So follow is very, very important. But yesterday I hear many things like in Odisha, going foreign or abroad. That is very poor. That is Odisha people. But in the neighbor Andhra state, is very common. And the importance of one thing is related with the importance in the management marketing. Suppose we are thinking a group for a daughter or for a bride. What the parents will think first? Which profession is most important in the market? In our Odisha concept, we say a doctor means first prepared. But in Andhra, we see one is working at Europe, US or UK or Germany or somewhere, they first priority abroad. So, important thing is awareness to grow. So, what awareness we got from this program, please use that. All the PPTs what we received from the different resource person will say in the FDP group, because a temporary group, no person will tell you hi, good morning, good evening, anything, just stay connected minimum for a week, after that we dissolve. So PPTs will say the mail IDs and contacts of different resource persons will say, so that it will be useful for the participants. So for that sake, hope all might have gained at least something from the program and use that in the coming days and future for the benefits of the own or for the benefits of the students. So it became little lengthy, time taking, but it was my responsibility also to say something in the report of this program. Once again, my hearty thanks to one and all for this. My best wishes. Thank you all. Jai Jagannath. May God keep you all healthy and happy. Thank you.
Thank you, sir, for the report and all the blessings. Now, I request the delegates from the different colleges if they want to share their experience on this 1 week and some program, and that will be, please take the stage.
No, they must require from the outside. Okay, our inside also. Yes. Our alumni.
Respected dignitaries on the dais, along the dais, esteemed faculty members, and my friends, teacher friends. A very good afternoon to all of you. I want to thank the organizing committee, chairperson, sir, then the member, the management team, principal ma'am, and the esteemed faculty members for organizing 1 week FDP PCI sponsored and recognized by IPGA at Royal College of Pharmacy and Health Sciences. So it was a very meaningful session, and the eminent speakers, those who have given the online sessions and offline sessions, it has touched the knowledge, our knowledge, and we came to know about the course outcomes, the CO, PO, the Bloom taxonomy, their perspective, how the AI should be implemented in pharmacy education, research methodologies, and we were able to know how to find a patent and how the research can be very easy and the things like the ethical considerations or anything which are related to our research field, which are related to educate our students, that it has been enriched to us. So I thank all the organizing committee members for making us a part for this week. Thank you. Thank you, one and all.
For sharing your views.
Very good afternoon to the respected dignitaries on the dais, and the organizing secretary Dr. Ranjan sir, and all the members who are actively participated in this program of 1 week FDP in RCPHS. And all these sessions are very nice, and the resource persons are excellent, and it is really helpful to all the faculty members for developing their skills in teaching and research. But one thing I would like to say, artificial is always beyond the natural. And one proverb also I am thinking, I think teachers are the life book, books are the dead teacher. Artificial intelligence is never be artificial. It is man-made. In this context, one example also what I observed during the 1998 to 2000 era is in cricket. I am giving one example. Sandborn, Bradley, and demonstrate the best bowlers. They are putting the ball in a high pace of 140 kilometers. Difficult because India is popular for batting skills. Difficult for Sachin Tendulkar, Dravid, like that. Batsmen are there. What BCCI has done, bowling machine came and control the speed of 150, 155, 40, and the bowlers in next matches observed that they are right. The intelligence in the same pace they are coming to pitch and arm action are same and do ball speed is increased. Then it is difficult and then they will get. It is the man-made intelligence, whatever we are searching in Google or artificial intelligence in chat GPT, AI, etc. It is not beyond than the human being. We are the creators, we are the creation minds. So don't be disable your mind, open your mind. If you are totally obsessed with the intelligence, artificial intelligence, chat GPT, Google, etc. After some time, your brain will come just like a closed umbrella, not a open parachute. And if it is a closed umbrella with our exit parachute, if we jump, we will die. Thank you all, and really nice arrangement of RCPHS. But I observed many of the things I attended, but I have seen really nice arrangement, hospitality, hosting, and all the things. With all the participants are satisfied in a larger extent, and we hope and we request to our chairman, Dr. P. Prakash Rao sir, Principal Ma'am, Dr. Musarani Madam, and Organizing Secretary Dr. Ranjan Kumar sir, and what we observe, all the faculty members are actively participated wholeheartedly and made it successful. We hope further such kind of programs in RCPH system. Thank you all.
Thank you, sir. Anyone else?
Good afternoon, everybody. Respected dignitaries and my teachers, since I am here with Royal from 2003, now also my kids are sleeping with fear to talk in front of my teachers. I don't know how Sunita has talked, but now also I am sleeping. I don't know where Sudhir is. Sir told that you go on the stage and tell, but it's very hard for me. Now the students are different. If I will say about me or Sunita, our batch, we are tried of our teachers like anything. And also, me myself, from 2003 and 7 batch, we are very happy and we are very blessed. We got the best teachers in the world. Either Sudhir sir, either Ranjan sir, and everyone, we got the best teacher in the world. And from them also only we learn many things. If I talk about today's FDP, the big law FDP, organized by Royal College. Till now I am saying that this is my college. Now today in the morning only I am telling suddenly that this is my college. I never felt like when I'm coming here. I'm a student only, and still I got many things to learn from the FDP and the strategy. And sir told the right thing actually, Rana sir. Yeah, the artificial intelligence entered the world. We have to learn, definitely we have to change the world. But no one can change the teachers. The thing we learned from Sudhir sir, by either scolding or learning, that will not change by activity. That cannot be changed by AI. The first thing, the Anjan sir classes of activity. So that cannot be changed. Yes, we learned the practical system was very much informative and we learned many things and definitely we will set the things in with students and thanks the management committee of Royal College for organizing this event and thank you Dr. Anjan sir and all of my faculties over here for organizing this one and enrolling me in this. Thank you.
Thank you for the words. Thank you. Thank you for all the efforts from different colleges. Hopefully, we have justified your expectation. Okay, sorry. Respected the Prakash Kaur, Secretary, Sir, Principal, Dr. Usharani, and friend, Dr. Anjan Kumar Mahapatra. I was speaking over here as a participant, not as a faculty of this institution. Because I've been participating since from 5 to 6 to 11 yesterday. Actually, here I see that most of the resource persons are either my students or my contemporaries. I'm very happy. I've been enlightened with my students because they gain more knowledge than me. I'm very happy what teacher wants. Teacher always wants the students should prosper. So coming to this theme, I'm coming with a really my student. Truth. Actually, we are creating a. That means we have to dictate the things not dictate us. So that we have to understand. And coming to the other things, basics, advances, and all the things. Most of the topics covered by most of the resource persons. And coming to another thing, recently, the first day, our speakers given one idea
talk on NPC 2026. Actually, this NPC 2026, this is the second draft. The first draft because of different suggestions made by public. Then second draft. The second draft actually what NDP is saying, National Education Policy. What say multiple entries, multiple exits, A, B, C, all the things covered. Suppose if you think about only pharmacy curriculum, only modern system of medicine, it is not possible to give either multiple entries or multiple exits. That's why this NPC 2026 is covering Indian system of medicine, that is Indian knowledge system of medicine, modern system of medicine, but some people are raising their voices in social media that I should be excluded. Actually, nowadays, the era is the era of integration and collaboration. So, keeping in that view, they made it that one. Of course, I already sent my suggestions, whatever the suggestions after reading that. I will not. Good. Of course, anyway, thank you very much for this opportunity. Thank you, Pananda. As the participants, I am telling all lecturers are enlightened very well. To me particularly, I am impressed very well in this session. Thank you very much. Thank you all. As highlighted to National Pharmacy Commission, we have posted this in our FDP group. We will make a Google form. So we request all the participants to give a feedback in the Google form about the National Pharmacy Commission. It's a request already we posted, taken a vote. Within 1 minute, we give the feedback, comments on this.
Any other faculty member from our college share something?
Good afternoon, everyone. Respected governing body members, Chairman Sir, Prakash Rao Sir, Secretary Sir, Secretary RER, Principal Madam Dr. N. Usarani Madam, Vice Principal Sir Dr. Kumar Sankar Sir, and Organizing Secretary Professor Ramchandra Kumar Bhadra Sir, respected senior teacher colleagues and junior teacher colleagues. A very good afternoon to all of you. I am Priyadarshini Parida, working as assistant professor in this college since 2019. And really, I first of all I would like to thank the organizing committee of the FDP for arranging very nicely and the smooth running of the events. As well as I would like to thank the managing, the governing bodies for their kind support and giving the opportunity us to learn the teaching pedagogy, basics and advanced in the research and application of AI in the pharmacy. So really it is very, very effective and we learn much more things. After a long time, I felt that I am now a student because I missed it, because during student time it is still good and I feel very happy. We also happy because in our college, like this type of activity, arrays will come across to know about many eminent resource persons, faculty members from different colleges,
And in the long period, that is 1 week, we spent very nicely, very happily. And I also expect this type of program should occur frequently in our college. And some suggestions also I can say regarding the development of both physical, mental, and financial basis. Like this is the one topic. In further future, on this topic also we can arrange the FDP. And thank you. Lastly, I thank you all of you.
We'll now move on to the next session. That is the
Finally, let me have a small mathematics in this regard. We have spent, all of us spent so much time, so much efforts, so much cost. My friend is an auditor and in a chapter. See that in his view, how much is cost effectiveness of this program? How much is the time effort we have spent? Everybody, wholehearted, even the participant. Suppose somebody has come from distance or somewhere, they have stayed here for 7 days. 7 days spending, how much time, effort, mind, and cost, effectiveness, how much they are enriched, how much is they enlightened themselves. The output should be the profitable. Somebody has missed it, they should feel that we have missed a great program. And there should not be any dependence. who are just certificate ରହିଲି ମୁଁ ଭଲ କାମ କରୁଛି ଭଲ ଶିଖିଲି ପିଲାମାନେ ଭଲ ମୁଁ ଶିଖେଇ ପାରିବି ମୁଁ ନିଜେ ମଧ୍ୟ ବହୁତ ଆଗକୁ ଗଲି one time I have told that you have to find a new you in you ନିଜେ ଭିତରେ ନିଜର ଉଦ୍ଭୁତ କେତେ ହେଇଛନ୍ତି ଆପଣ ଜାଣିପାରିବେ so with effectiveness is most important self-confidence development is more important as somebody has told that natural intelligence is much more better than anything else so this is confirmed so everybody is happy spending 7 days here for academic fees that will be the outcome they misquoted the time is they are mistaken. I request Professor Sudhir Sir that from the beginning I told that please take out the glimpses and summaries. Make a answer and that will be
it's up to time. Okay, up to date. 2 days report only.
Okay, thank you.
Thank you, sir. As I said, somewhere the only investment that guarantees a profitable return is the time invested in learning. So, thank you. Now, I would request our governing body member, Sri Accredited sir, to share a few words.
Very good afternoon to one and all. Dignitaries of the Secretary, Second, Principal, Washington Administration, Secretary, thank you all for giving this opportunity to speak to us much related the grand program of last week. In fact, I feel very sorry for not making slides. For the first day, last day, really enjoying getting a very positive vibes from us. Very happy to see that it's a very good program. We suggest from our board and society to conduct more such programs. And we are really very happy to have the resources such as finance are, these are a lot of hope. And we are encouraging them to make some more useful programs which will benefit the segment as a whole, pharmacy or any other segment. It's a really good thing to start with the teachers enrichment program. And really the particularly they chosen the beautiful topics and the resources questions also. The selection was very meticulous, and I really very happy to enjoy the total week. I assure from our management, more programs of this kind will be in pipeline. And we really get good feedback coming because what some of the things you expect, and we are always with you. Thank you very much.
Thank you, sir. Moving on to the next section, would like to appreciate all the delegates for their participation, and for which I would request our dignitaries to come forward. And I will request Rajastri Ma'am. I will request G. Sai Chatri, Assistant Professor for the Pharmacy, to do that.
After the certificates and membership distribution, we join the launch. Then it will be followed by cultural activities or anything. Our time like that. Anybody interested on?
In-house consultant.
So,
A very good afternoon, one and all present over here. Now, I would like to invite our Chairman, Sir, Deputy Prakash Rao, Sir, and Secretary, S. Prakash, Sir, to facilitate the participants with certificates and mementos. Because topics becomes very recognized. I would like to invite Mrs. Shravani Pradhan to collect the certificate and the memento. Mr. Vishwanath Trustee, Mr. Anshman Gowda, now I would like to invite Mrs. P. Neeraj, Ms. Madhusmita Ekta, and Mr. Ravindran Rana to collect their certificates. Now I would like to invite Mrs. Swagatika Mondan, Mr. Manthan Kumar Arupal, and Mr. Sandeep Kumar. Sunita Patnaik, Ms. Arpita and Mrs. Ankala Anushka to collect the certificate and now I would like to invite Mr. Anup Kumar Patra, Mr. Satyanarayan Sahu, and Ms. Divya Sarika to collect their certificates. Mr. Varun and Maratha sir to collect the certificate. Amazing Secretary Anjan sir to file certificate. Mr. is for certificate. To facilitate the participants. Now, Professor Kumar Sir, please collect the certificate. Like to request Professor Kumar Sir, request Now I invite Mr. Ujjwal and Falkira to collect the certificate and now I invite all the certificates.
Next, I would like to invite Dr. K. Harikrishna sir, Mr. Burham Pani, Mr. Narayan. Narayan. Mrs. Kari Abdulati, Mr. P12X, and Mr. Desai. Our district organizing secretary, Mr. Ranjan Kumar Mahadas, mentioned the routine of fishing. Without his ability, would not have been possible. Thank you, sir. Now, I would like to invite our esteemed coordinator, Dr. Usharani, to kindly receive a token of appreciation, her sincere commitment and planning played a vital role in the successful conduction of this FDP.
May I request the faculty participants from the respective institutions to kindly collect the certificates and evidence on behalf of the participants who are unable to be present today here. Thank you.
Teacher, Professor Prasad Kumar Chaudhary sir, to please provide a vote of thanks for this program.
Respected Principal Madam Professor Sir, respected Vice Principal Professor Mr. Sir, and my brother Professor Anjan Mahapatra Sir, Secretary, governing body member Sri Sakar Sir, Dr. Bhagan Vishwan Sir, Principal Mr. Anand Sir, participant from all my senior colleagues. On behalf of the organizing committee and members of Indian Pharmacy, I have been given my responsibility to extend both of them. Going to be repeated. So I will take this opportunity to repeat the same thing. Each and every speaker on the dais for this benefit discussion and participant, nobody has missed a single opportunity to extend the thanks to the organizing committee. But to start with, let me say a statement. IPGA turned into action. After that, this FDP has been. I think this FDP to start with soon after the reopening of the institution. So during the break or during the summer vacation, we all tried to organize this with discussing and the stage to be the proposal by our beloved esteemed chairman, Dr. Brother, and I remember that day. Instantly, I told that we should have FDP, and immediately he said, yes, go ahead. And I'm telling you, some of you just decided PCA sponsored. Let me correct, it is not PCA sponsored, it is PCA recognized. We are associated ourselves with the Indian Farmers Welfare Association. It is not sponsoring FDP, though later on they extending and lending their IPGA to some extent, but this is not funded. But this has been conducted smoothly by the funding of our society. This society will president with none other than our chairman, Dr. Sridhar sir, engineer Ashok Padikar, Satyaraj sir, each and every member, they are so much keen towards putting this kind of programs and always extending their help in terms of both financial, mental, as well as physical health. You have seen this, those who we have seen the change, and our alumni members who have came back, like Sunita, Swagatika, and others. This was the class 2. Now it is a beautiful auditorium. How this happened? This has happened because of the vision and help and guidance of all our governing members, Prakash Rao sir, Srikant sir, and we have got this auditorium. Now we are feeling very happy to organize this kind of programs also. Thank you on behalf of all the alumni members, all the faculty members and all the alumni members, particularly I take this opportunity to thank you.
In this part, I recognize the unsung heroes who have day and night done this job. Ravi, Ravindrachari, Bala, and so many people.
Each and every
people. The generators, our all of these people sell generators.
Yeah, absolutely right, sir. Starting from the sweeper field to the end of the institution, everybody has done their efforts, and we have got this very delicate, colorful, and very pleasant auditorium. So the patrons who guided us to organize this FDP, starting from, though they are not present, I would like to take this opportunity to listen to Dr. Amit Nasa, President IPGA Central Body, Professor Arun Garg, Executive IPGA Central, and our state IPGA President, State Branch, Dr. Lalit Kumar Rao, and Professor P.R. They also helped us to organize this FDP group, helped us to organize some resource persons like as already told our ex-faculty member and my senior brother, Dr. Sudarshan Vishwa, he of Accredited Development, others are to invite the eminent resource persons. So let us take this opportunity to extend our gratitude and thanks to all of our patrons. See, this program has been conducted. We know each and every person has been involved. So as I told, it's a non-funded or non-sponsored FDP. So there has been effort by the financial committee. The financial committee, which was been headed by our vice principal, Mr. Sir, and along with the team, Mr. Sudhanshu Sekhar, they calculated this and they are still that accountability, like what we have invested, what we have what we have got, everybody is yes, sir. They will be showing that account and maintaining that. And then we have started with this. It goes to the brochure, preparation team, Professor Rajeshri Panikani, Mr. Rajesh Matia, this ICT. They started delivering about this information in social media, regarding hard and both soft copies of the brochure and circulated it to all social media, and we have got this information about this activity. Soon after this has been circulated, the next step was the registration. So the registration of the candidates, and as usual, we know we have got working hard leader among us. Professor Madhuswati Khandai Madam, and along with this her team, Mrs. Priyadharshini Parida Madam and Mrs. Archana Pandey Madam, they really arranged this registration. On the first day, we have observed every day the delegate of participants have been very heartily welcomed and handed over this registration and all this. So thank you to the registration committee members. Yeah, this till now that I am standing here and delivering, we are organizing this, but we have observed 3 or 4 people are continuously handling all the sessions since from inauguration till now. So those dynamic, young, energetic team for handling all this activities, starting from hosting, handling, and delivering all the things, the team of and impressed along with all our team members. I would like to thank Mr. Ashish Sahu, Mr. Rajiv Khan, Ms. Vajasi, Ms. Aishuti, and all other coordinators. I may be missing 1 or 2 more names, but it really your enthusiasm, your energy is still inspiring us to continue like we people are doing. Though we are not that much older. Thank you for this organizing all these lessons. And this session was maintained by the team of session maintenance, Mr. Santosh Patnaik, Mrs. Sweta Mr. Madam. They really done the pause part, they will maintain very well. And with FDP, we have organized with the intention to deliver some latest information about the basics, advancements in research, application of AI, and as a teacher, how we develop by learning through pedagogy. So we have arranged all the resource persons. But these resource persons have been delivered very nicely, all this through the PowerPoint, and we are continuously having this YouTube live. We are sharing the link. You can go back, you can see back and memorize. You can have the memories to be recalled again. So for this maintenance of scientific sessions and all this, the credit goes to scientific committee members. headed by Professor Sudhir Kumar Sahu sir, Mr. Sharath Sahu, Mr. Rajesh Bhatia, Mr. Bharti, and again I am telling on my personal behalf and on behalf of all the coordinating committee members, let us give a large round of applause to this team particularly. And we are really highly impressed with the technicality handling, how to task it, and all the they were appreciating about this live telecast and all of this. So credit goes to this team of Sudhir sir, Tarango and our technical Shelly, Rajesh and our team of young members. Thank you for organizing this scientific session so nicely. I will be failing in my duty if I will be not highlighting this lab and software setup team of Professor Ghanshyam Pandey sir and Mahapatra for arranging all the setup during also these hands-on training sessions. Yeah, everything is okay, everything has been appreciated. But empty stomach we do not do anything. I know. Also, we are getting feeling hungry and we are getting already empty stomach. But I hope you will enjoy this and you will be feeling very happy to appreciate the team of person. Professor Rasulanan Sarvedi sir and Mr. Narayan Pradhan. What I got between on my personal game and we have also tested all of my delegates and participants. The food was very, very nice. So, because of our problem with that is why that saying, I'm the guy who should be one. We do not optimize on quality, whether it is teaching or whether it is any other session or you are not on the food. So, credit goes to Professor and all the cooking staff members. Thank you for serving us this delicious food. Though we were glad to take that food at time. Yeah, all have been recognized. Particularly two things I left intentionally. Organizing everything and socializing it that we have organized. With the utmost care of hospitality and efforts to feel everybody that is members story. So that goes to our hospitality team. The hospitality committee headed by Professor Prasad Patil sir and along with Dr. K. Harikrishna garu. I like because he is so enthusiastic and along with Professor Patil sir and Harikrishna garu, they are so busy in everything, all these guests to come and take their pleasant stay, even they arrange some investment. Thank you, sir, for making our resource persons to feel that they are not available only, and thank you for all these hospitality arrangements. Yeah, that is very nice. And that really goes to hospitality. We have been recognized, and I am very proud to say that this program has been covered by minimum 20 written media and more than 10 non-printed media. Since from the inaugural session till now, it has been covered and well circulated throughout the state and even outside the state, and it has been recognized and read by each and every viewers and their views. So this praise publication of the information in press, printed press and in social media, it has been taken care. Very happy they have done their responsibility, and the credit goes to Mr. Asim Baran Maniknaikar, Mr. Rajesh Kumar Bhatia. Along with that, I am just clubbing another committee, that is photography and reporting by Professor Odisha Patel sir and Mr. Bharti. Everybody has observed and everybody has seen their work. how every day he posting the photograph and making it an album. So each and every participant has gone through the continuous work done by Mr. Priyalan and guided by Professor Pasa sir, that they have captured each and every movement of this FDP and well displayed in the respective blue set social media. So thank you for making this message captured so that later on we can change that again.
You make it actually.
Yes, obviously. So it's time now to end up this valedictory session. But I will be failing in my duty one more time if I am not considering the name of Professor K. Aparna Chaudhary sir and Ms. Arundhati Behra along with Rosalie madam. Whether we have done very nicely arrangement or not, feedback has given by whom? That feedback part they have taken care, particularly Ms. Arundhati and Aparna Garu, along with Rosalie madam, they have taken care of all this feedback, assessment, and all these things. Thank you, sir, for giving us the feedback back to us, what we performed or not. Yeah, as the list will continue, I know all are feeling angry and somewhat all is getting. I will not take much time, but exactly I am feeling in my group. I am not extending my heartfelt thanks and gratitude to each and every participant. On behalf of the whole organizing committee, members and our governing body members, management, and each and every family providing knowledge of farmers, I would like to extend my heartfelt gratitude and thanks to each and every participant who came outside, spending your valuable time and getting this valuable information simultaneously. So I would like to request all to give large round of applause for yourself. Thank you.
Sir, you forget your department.
Yeah, thank you for opportunity. So self-efficiency one may not be common. So it is a big mistake, being the organizing secretary, not to honor and appreciate the logistic team, which is headed by Professor Prasad Kumar Chaudhary, in essential Mr. Gargi Arundhati and Subha Pandey. This logistics part is very important. Means in the company also, this department is very important. And if you are out of place, in our banks, many brilliant are there. Even Dhananjay Pandey, he was ranked 21 and 8 PhD. Manoj Kumar Padi, his friend, Prasad, is below in things comparison. But in our page, even if we compare our seniors also, he is getting nearly 1 crore for annum. Working as a logistics head or Dr. Reddy Saburi. Very important. I appreciate Professor Prasad Kumar Chaudhary and the team logistics for helping in all aspects. Last but not least, being the organizing secretary, on behalf of all the teams, please excuse if anybody has any inconvenience or trouble or problem during this monthly program. On behalf of all, I express my excuse. Please bear with. It was your program and it was organized by your friends or family. Thank you all. With this, with the permission of the president, I would like to declare this valedictory program is adjourned. And all will join for food launch with the left within Hungary. And after that, we will do cultural program for everyone. Okay, 15 minutes cultural program will do. Okay, that's I would like to request all the participants to go for lunch. And